welcome all of you to today's special lecture series of Lecture 1, Carcinogenesis and Immune Modulation of Cancer. Department of Studies in Genetics and Genomics, University of Mysore. I request Professor Dr. S.S. Malini Ma'am, Chairperson, Department of Studies in Genetics and Genomics, University of Mysore, to grace the dais. I am glad to invite Dr. Vinay Muttagi sir, Consultant Radiation Oncologist, Bharat Cancer Hospital, Mysore, onto the dais. Now, I request Dr. Kiran P.K, Assistant Professor, Medical Oncology, JSS Medical College, Mysuru, onto the dais. Prayer should be the key of the day and lock of the night. So, I request Sinchana for invocation song.
गजमुखवन्दे सुरे करुणिसि कायो गजमुखवन्दे सुरे करुणिसि कायो गजमुखवन्दे सुरे करुणिसि कायो गजमुखा
Good morning, one and all. Indeed, it is a great pleasure for me to invite two dignitaries on the dais, Dr. Vinay Muttagi and Dr. Kiran, on behalf of University of Mysore, Department of Studies in Genetics and Genomics. Heartily welcome to you, sir, for this occasion, a special lecture series. I request Senior Professor Subramania sir, and Dr. Chaitra, research scholars, students, everyone. You must be knowing about cancer. What is cancer? In a layman language, it is cells have gone mad. That's all. A very simple thing. How they have to behave? They don't behave. Okay, sometimes how our students behave, no? That time I'll be thinking what happened to them. Okay, so no control, high cellular proliferation, and what they're supposed to do? They will be doing something else. The defined work will not be done. Okay, Cowdrey has given this definition. Cells have gone mad. How the mad person behave, lost control on entire thing, on his mind, same way he behave. And what's the reason? You must be knowing today, because there's a change in our lifestyle, diet, habit, the incidence of cancer, the exposure to carcinogens that resulting in carcinogenesis. But still, we'll be knowing our body is amazing thing. So, which is the wonderful thing in the world? There are lot many you can list. There is a Taj Mahal, there is a wonder, China Wall, wonder. But our body is a mesmerizing thing, because each and every action at molecular level you must be knowing the perfection to the core. If there are any damage, immediately body will recognize. And there is a DNA repair mechanism. What a fantastic mechanism we have. Every day you eat the barbecue, all the food additives, coloring agent, gobi manchuri, pani puri, all these things with Ajinomoto, coloring agent, food additives, pesticide, insecticide, fungicide. So we eat toxins, carcinogen, we drink a carcinogen. In spite of that, so we had not been to Bharat Cancer Hospital till now. How come? Because our body has a defense mechanism. Defense at different level. Okay, so DNA repair mechanism is there, apoptosis is there, too much suppressor genes, lot of suppressor, dozens of suppressor genes. Okay, so today both the dignitaries, they will take you to the world of cancer in general, and immunology and immunotherapy. And Dr. Vinay Muttagi is a great orator, both of them. Today it is a feast of knowledge for you. I don't know whether you have had your breakfast or not. Definitely you have a breakfast for your brain. Okay, with these few words, once again I welcome to you sir on behalf of everyone. So our students are like a inert element. I don't know how to ignite them. I'm trying, but I don't give up. That is not my nature. One or other day, definitely I will make you very active and productive and trying. I never stop my attempts. Whether it is a Ghazni Muhammad 11 times, 12 times invasion, no problem for me. I keep on doing my work. Okay, because that is called the Karma Yoga in spirituality. Okay, so with these viewers, once again I welcome to you sir. And now I request our students to introduce them to the gathering. Thank you.
Thank you, ma'am. Now I request Kavita for the introduction about the guest speaker.
Dr. Vinay Muttagi has over 9 years of experience as a radiation oncologist across various hospitals in India. In this short span, he has achieved exemplary precision in performing various complex advanced cancer therapies like stereotactic radiosurgery using cyberknife and shooting technologies, stereotactic body radiotherapy, volumetric modulated arc therapy, intensity modulated radiation therapy, dimensional radiotherapy, intraoperative radiation therapy, true beam radiation therapy, and radiosurgery. He has also enormous experience of brachytherapy in carcinoma cervix, endometrium, and esophagus. The doctor has special interest in brain cancer, breast cancer, cervical cancer, awareness about cancer and its early detection. Dr. Vinay is actively involved involved in many public awareness and cancer screening camps across Karnataka. After achieving success in most of his cases, today he is one of the best radiation oncologists in Mysore. Dr. Muttagi finished his MBBS from an esteemed institute, Sri Devaraj Aras Medical College in Kolar, and completed radiation oncology in Karnataka's Indian Cancer Center, Kiran Memorial Institute on Oncology, Bangalore. He has acquired training from some of the most renowned hospitals like PGS. During the last 9 years, he published many research papers in various peer-reviewed journals. Currently, he is working in radiation oncologist at HCG, Bharat Cancer Hospital, Mysore.
So over to you, sir.
We have chosen two different topics. I'll be covering on carcinogenesis and Dr. Kiran will be continuing on immune modulation of cancer. I will not get into too much of in-depth about genetics and all. I'll try to introduce you to cancer and the genomic part of the cancer. And how did that evolve in malignancy and what translation that we got knowing the entire human genome project and its application into malignancy. To begin with, if you start with, probably you might have, all of you might have gone through this images multiple times as a genetic students. Probably if you see from the left hand to the right hand, this clonal expansion or clonal theory which was tried novel, which clearly depicts that how malignancy arises from a cell. All of you know that the entire human body is made up of millions and billions of cells, and group of cells which are destined to function in a specific manner are designated as organs. Say, liver is made up of a group of cells which are called as hepatocytes, which are meant to function the metabolism of the liver functions of liver. All these cells which are organized as a group of cells or which are asynchronous with adjacent cells, which are meant to function in a specific manner, they are all interregulated, and that entire functional activity is hidden in DNA. That is, the entire biological information of an individual cell is the DNA. So, whenever a cell tries to function, whether it is in an autocrine mode or a paracrine mode, most of the times what happens is that cells get signals or in a layman language that from the DNA to perform in a specific manner. And the individual cell has a lifespan where the entire bunch of cells will sense whenever the cell is going through a senescence or whenever there is a cell deficit which is happening within the system. And that signals will be sensed by the stem cell and where they will procure more number of cells into the system. And that is how the cell cycle is regulated individually. And most of the times it is completely a controlled system, and which has lots of growth signals, growth receptors, transcription signals which are happening at the molecular level to control this cell cycle or cellular growth or cellular duplication. Whenever such duplication is happening and any one cell which picks up signals or starts functioning in an autoctonic mode without any growth signals or without any signals coming from the outside, such stem cell or such clonal cell which starts acquiring features of self-sufficiency where it starts replicating on its own. Such replication creates number of cells, and like such clonal expansion would acquire multiple genetic alterations within the system, and such form of clonal expansion which forms a mass or an ulcer is called as malignancy. So as I told you, cell division which is leading to cell duplication, or it is a tightly controlled process which involves multiple growth factors, growth factor receptors, signal transduction proteins, and along with that, as I said, that at the moment the cell is sensed to be senesced, it undergoes a programmed cell death, which we call it as apoptosis, and whenever there is a damage, as Madam was telling, that such damages are identified, and if the cell is still viable, it will be sent back into the system by DNA repair. Any cell which is during the process of duplication, which will undergo a genetic screen, what we call it as any number of cell duplication which is happening every time, with the entire genome would undergo a screening process just to identify, just to have checked that the entire genome is functional and there are no abnormalities detected. The moment there are no abnormalities detected, then only the checkpoint allows the cell to enter the replication cycle. Otherwise, the cell is detected defective, and that would lead to cell death, that is called as apoptosis. If it is feasible, then the cell is sent back to a repair, and from there to the cell cycle back again to the system to function in a normal way. Cell which escapes this sort of a screening process would avoid apoptosis, would avoid the cell death, would avoid the genetic repair process, and that would get back to the cell cycle system. A defective cell or a defective genomic defective DNA which get back into the cell system would start functioning in a potential way, start duplicating without any controls from the outside, gain self-sufficient signals, and that transforms into malignancy. The same again in the pictorial representation that cancer is characterized by abnormal growth of normal cells, which arises from the loss of control from the normal developing cells. There is a huge imbalance, just on a blanket statement, there is definitely a huge imbalance at the micromolecular level. Just to remember that it is uncontrolled cell growth, at the same time avoiding apoptosis. That is something where the cells are growing independently, at the same time they are devoid of cell death. I mean, they have escaped from the regular screening of apoptosis. This is the picture of cell cycle. Say a cell which is going through a duplication. The moment a stem cell senses that there is some lack of cells in the system, it gives a growth signals to the stem cells so that it can pick up a cell from a senescent quiescent phase to that of a G1, I mean growth phase. From there the cell enters a duplication process. This entire cell cycle is completely controlled by cyclins and cyclin-dependent kinases. I think I'm sure that everybody would be aware of cyclins and cyclin-dependent kinases, and they, I'll just brief it off, just to get an implication that how we evolved when it comes to how did we understand that how tumor growth happens, and how do we sequence our treatment based on this cell cycle. Say a quiescent stem cell which is picking up a growth signal from outside would sense that the cell has to duplicate. It starts recruiting cells from the G0 phase to G1 phase. From there to cross G1 to GS phase, it will activate cyclins. The major cyclin which starts rising in the late G1 growth phase is cyclin D. So along with cyclin-dependent kinase, CDK4. So the combination of cyclin and cyclin-dependent kinase 4 would phosphorylate retinoblastoma gene, and in that the phosphorylation of that would lead to the subsequent events of transcription where the cell will start duplicating, making this duplicate copy of DNA, and then processing from G1S to G2 and then to mitotic phase. So this entire process of cell cycle and the entire process of cyclin and cyclin-dependent kinases, there are multiple cyclins and cyclin-dependent kinases which would shoot up in every phase of cell cycle. The major one would be the cyclin D and cyclin E and cyclin B and cyclin A, and the dependent kinases along with the cyclins in each phase of the cell cycle. The majority of this cell cycle is controlled in a very systematic pattern where they have a growth promoting proto-oncogenes and growth inhibiting tumor suppressor genes. That is a misnomer, basically, that doesn't suppress the tumor per se, it keeps the cell cycle under check, that's all. And the program cell and DNA repair. As I have told that any cell which is undergoing the cell duplication process would get the signals from proto-oncogenes just to duplicate it in double copies. And if once it goes through the duplication process, it goes through a check process of P53 mediated check process. And if it is deemed to be viable, then it goes through the system for functioning. If it is deemed to be which is not a viable cell or not, if it is a defective cell, it would undergo a DNA repair. In spite of DNA repair, if it doesn't come back to the system, then it will be sent to program cell product. With this cell cycle, what happens? A normal cell getting a signal from outside to undergo duplication. Similar way, any cell which is being damaged by external or environmental factors like chemical agents, radiation, environmental factors, or the viruses would pick up some DNA repair. And the moment a cell gets a signal that the DNA being repaired and cannot be processed further for the cell cycle, that would be labeled as failure to repair the DNA. At this process, either it is that if the DNA repair mechanisms are intact within the cell, then probably the DNA repair happens and the cell goes back to the system. If there is no good DNA repair mechanism system, then further replications of the same DNA or damaged DNA would acquire multiple mutations, and these mutations causes genomic instability at the somatic levels. And these start activating growth promoting oncogenes, inhibits all the tumor suppressor genes, and causes alterations in the regulatory process of apoptosis. Mean to say, it will create a system in such a way that it can replicate n number of times, avoiding all the inhibitory checkpoints. The moment it gains that self-sufficiency, then unregulated cell proliferation and multiple clonal expansion. Once the moment the tumor cell picks up this clonal expansion in a different way, it starts creating a microenvironment in and around. The basic nature of a tumor or malignancy is to grow at the place where it starts, and from there it's try to metastasize to the other parts of the body. So invasion and metastasis is the hallmark sign of malignancy. And to do this, the clonal expanded tumor cells would start creating a microenvironment in such a way that it will start having its own blood supply, its own oxygen, its own nutritional supply. In that way, it creates angiogenesis. It escapes from all the immunity, that is, the body resistance mechanisms to avoid this malignant cells. The moment it escapes from the body's immune mechanism and starts gaining additional mutations, that is when the tumor starts progressing. Most of the times in our clinical practice, all these things which is happening at the wouldn't come into the picture at the early stage, and we start noticing only the tumor when it has crossed all this and it started becoming, when it is already there in the progressive stage. There are lots of research which have happened to diagnose malignancy at the earliest. Just to give you a picture, as I told you, this entire process is at the micromolecular level, and for a cell to be recognized or cell to be seen in anything like be it imaging, just like an X-ray or a CT scan or an MRI, it would grow up to a size of a gram or something. It would take at least definitely 3 months. That is what we call it as tumor doubling time. The tumor cells does not double more than that of a normal cell. You mean to say the duplication rate of a normal cell and the tumor cell would remain the same. Only thing is that the tumor cells doesn't undergo apoptosis. They have that uncontrolled growth capabilities at the same time, avoiding apoptosis. In that way, the tumor cells to grow, to become malignant, to appear on an imaging or to get diagnosed clinically or become clinically apparent, it would take at least 3 months. And that is where we probably, most of the tumors, some of the tumors might be very dominant, some of the tumors might be aggressive. That depends on the histology. Again, that does not get into that depth. What I'm trying to convey is that with this micromolecular things which are happening at the earliest, probably if we are able to pick it up at the earlier stages, that is when we can really change the clinical picture of tumors. So as I told you, this malignant transformation would definitely require self-sufficiency for the growth signals. That is, the external growth signals which are coming might be altered, and then they start having a self-sufficient growth signals where without having an external stimuli, the tumor cells start replicating by themselves. Insensitivity to growth inhibitory signals, the way normal cell has a growth signals, similarly they have growth inhibitory signals also. It is a switch, on and off switch. The moment stem cell realizes that the further growth signals are not required or the further duplication of a cell is not required, it cuts off all the growth signals and sends back all the viable cells to the quiescent phase. So in that way, all these malignant cells lose that growth inhibitory signals and they start duplicating without any inhibitions. Evasion of apoptosis, that where there are lots of molecules which are involved in which are pro-apoptotic and anti-apoptotic. Most of the times, malignant cells will have that inhibitory actions of anti-apoptosis will be lost. And defects in DNA repair, there are lots of normal genetic disorders which are associated with defects in DNA repair mechanisms. Similar with the DNA repair mechanisms where damaged DNA or an acquired somatic mutation cannot be rectified and sent back to the cell cycle pool would definitely lead to malignancy. Limitless replicative potential, sustained angiogenesis. As I told you, the moment it starts duplicating, it creates a microenvironment because the moment that the tumor starts growing in a similar fashion, it should get its nutrients also to the blood supply. In that way, it starts creating its own blood supply through a sustained angiogenesis and ability to invade and metastasize from one place to other place. This is an example of cyclic and cyclic-dependent kinase, how you can act, and the multiple pathways which are involved. The moment this, if you see this is cell membrane and that of a nucleus, the moment the cell receives the signals, the growth factor signals, be in terms of platelet-derived growth factor, lots of number of growth factors are there. Just to give an example, like fibroblast growth factors or platelet-derived growth factors, the moment the growth signals come and it gets interlocked with the growth receptors, the signal translation starts happening at the inner surface of the plasma membrane. What happens? It will start activating the cell for the synthesis or for the release of cycling, the initial cycling, where it starts recruiting cells from the G0 to the G1 phase. The moment it is being recruited, then the cell cycle starts progressing from G1 to S and then to S to G2 phase. All this interlock mechanism has inhibitory signals too that are cycling. All these cycling dependent kinases can be inhibited by the external sources. At the same time, if the cell is found effective, the cell would automatically activate the apoptosis inducing factors, where the apoptosis inducing factors come into the picture and they start breaking the DNA in terms so that cell will go into apoptosis, that is non-inflammatory cell death. So as I told you this, all this is a completely regulated process, and at all the levels, at growth factor levels, at the receptor levels, at the signal transduction, all the signals, the moment the growth factor interlocks with that of the growth factor receptor, the signal transducers come into the picture where they transfer the signal from the external surface to that of the internal nucleus. So this signal transducing proteins, and once the nucleus receives the signals, the DNA starts replicating. All this is completely controlled by proto-oncogenes and tumor suppressor genes. The most commonly altered proto-oncogenes, what we have come across all these days, I've just labeled it in a table, and I'll not go to get into too much detail about it. Just to give you a hint that there are proto-oncogenes which will stimulate a cell so that it can replicate. At the same time, we have proteins which are completely involved in the signal transduction. From there, the nuclear regulatory proteins, and from there to the cell cycle regulators, as I told you, cyclins and cyclin-dependent kinases. The way proto-oncogenes and oncogenes are completely, you can call it as sisters, where the proto-oncogenes in the normal cell would help it to replicate, whereas oncogenes are the sister patterns of proto-oncogenes, where they help malignant transformation of a cell. Whereas tumor suppressor genes are physiologically, they are the genes which regulate the cell growth. This is a misnomer, as I told you before that tumor suppressor gene does not mean that they will suppress the tumor formation. It is basically to regulate the cell growth. You mean to say these are brakes basically, where say if you are driving a car, tumor suppressor genes are the brakes where they help to control the cell cycle. Whereas proto-oncogenes are the accelerator. The moment you start pressing it more and more, the car will go it in a faster way. I mean, like the duplication happens in multiple number of times. The parallel oncogenes are just the parallel proto-oncogenes which are seen in the tumors rather than the normal cells. So as I told you, it's a misnomer. There is a failure of growth inhibition is a key event in carcinogenesis. Loss of function of these genes, especially the tumor suppressor gene, the moment the tumor suppressor genes are mutated, that is when the tumor malignancy happens. They apply brake to the cell proliferation and they are products of the genes. Everything, whatever we are talking about, is all about the product of the genes. These genes act by coding growth controlling molecules of the growth factors. They control mitosis and the biochemical process which happens down the lane, governing the growth and differentiation. Similarly, as I told you, the proto-oncogenes which are starting from the growth receptors to that of the molecular level of nuclear regulatory protein. Similar way, the tumor suppressor genes also there from the cell surface to that of the nuclear level, like transcription factors, cell cycle inhibitors, signal transduction factors, cell surface receptors, and regulators of cellular response to the DNA. The major ones are R2 and P53 gene. So these are the two major genes, tumor suppressor genes, which play a huge role in malignancy. We'll just get in depth about RB gene and P53 gene, and all of you must be knowing that it's called as guardian of gene. So these are few other examples of tumor suppressor genes which are happening at the level of cell surface, in the surface of the plasma membrane, cytosol, cytosol, nucleus. The two major ones are P53 and retinoblastoma genes. So coming to retinoblastoma, I think till now I'm being clear.
Please continue.
Yes. And retinoblastoma gene. As I told you, the moment the cell receives a signal from an outside through a growth factor, say a proto-oncogene is sending a signal to a cell to duplicate. The moment that the growth signal comes and sits on the receptor and the signal transducing protein starts acting from the inner surface of the cell membrane and takes the message to that of the nucleus, and where the nucleus starts replicating, DNA starts replicating. So the quiescent G0 phase of a cell will be recruited into G1 phase, and all the growth factors, as I told you, play an important role to send a signal for the cell to replicate. Once this is activated, they start recruiting CDK, that is cyclin-dependent, cyclin D and cyclin-dependent kinases. This combination of cyclin D and cyclin-dependent kinase goes and phosphorylates retinoblastoma gene. Most of the time, what happens is retinoblastoma is in a hypophosphorylated state, which forms a tight junction with an elongation factor. I don't have a picture of that. This retinoblastoma forms a tight junction with that of an elongation factor 1. The combination of this would inhibit DNA duplication. The moment this is phosphorylated, the retinoblastoma gene dissociates from that of elongation factor 1, where this elongation factor starts recruiting histones into the picture, where it starts, the moment the histones come into the picture, the DNA starts replicating. So there is a tight junction between this cyclin-dependent kinase, cyclin D and cyclin-dependent kinase with that of the retinoblastoma. Once they cross this checkpoint, the moment cyclin-dependent kinase activates the retinoblastoma gene, then there is no comeback. The moment the cell starts to process this stage, then it is completely gone into the cell cycle and start duplicating. So this is a major checkpoint. The moment we have mutations which are happening at this retinoblastoma gene, be it in any form, if it is not able to form a tight junction with that of an elongation factor, the elongation factor is already free, it will start recruiting histones into the picture and the cell will get into the duplication process. If this hypophosphorylated form of retinoblastoma gets a self-sufficient signal where it is hyperphosphorylated throughout the phase of cell cycle, where there is no activation required from cyclin D or cyclin-dependent kinases, cell gets into duplication mode. So any form of mutation which is happening at the first level of cell cycle, that is with the retinoblastoma gene, would definitely push the cells into the duplication, and that is when cells start replicating in an uncontrolled way. So most of the malignancies what we encounter in our day-to-day life would have some sort of retinoblastoma gene mutations which are happening for a cell to develop into malignancy. Once we cross this checkpoint, then as I told you, it continues in cancer. At the same time, this the other regulatory checkpoint for the cyclin D and cyclin dependent kinases is the inhibitory growth signals which are coming from CYP family, which are completely again interconnected with that of apoptosis. Where this CYP and KIP family are E4 regulators, they will have a constant check on cyclin dependent kinases and cyclin D, where they will be sending inhibitory signals all the time to the cyclin D, such that it doesn't have a surge during any phase of cell cycle. The moment if there are any mutations which are happening at the inhibitory signals, say there are no inhibitions which are coming from the CYP family or E4 family, then the cyclin D and cyclin dependent kinases will not have any inhibitory signals coming. So there is a surge of cyclin D throughout the phase of cell cycle. That is when it starts phosphorylating retinoblastoma gene. So anything to do with growth factors, where the signals are coming uninterruptedly from the outside. Anything to do with the retinoblastoma gene, where retinoblastoma gene is in a state of completely hyperphosphorylated form throughout the phase of cell cycle. Anything to do with the loss of inhibitory signals, where there is no inhibitory signals coming and blocking the cyclin D. So cell cycle is not under check and the cells are replicating at uncontrolled way continuously. The similar picture, same picture I have dissected it in a better way. This is a cyclin dependent kinase, this is a cyclin D. The moment cell cycle gets a signal, these two will be raised. Before the signals are these two parameters are on constant inhibitory signals which are coming from P16. So anything loss of P16, anything which has a self-sufficient signals from retinoblastoma, keeping it in a hypophosphorylated form would result in malignancy. The other important one is P53, other immunosuppressor gene. Well, what I would say, suppose, as I told you, a cell which is being damaged by any of the environmental factors, be it in terms of chemical carcinogens, or radiation, or viruses, the moment cell recognizes that there is a DNA damage, the P53 system is activated. So what happens? P53 has a transcriptional activities which is happening to P51 and GAD45, and that P53 causes this cell which is being damaged to be arrested in the G1 phase before even it enters to S phase, where G1 S checkpoint happens and DNA repair enzymes are activated and the cell undergoes a DNA repair process. Say this entire system of P53 is defective. There is a defective DNA which is already there in the system, but it is not able to elicit P53 response because P53 is already being damaged because of any sort of mutations which are happening, be it genetic or germline mutations or somatic mutations. So the moment P53 is not activated, this defective cell would not undergo this DNA repair mechanism and it will be pushed into the cell cycle as a defective DNA itself. So the moment that defective DNA enters the cell cycle with the consequent or subsequent cycles of duplication process, it will acquire multiple number of mutations throughout the process. That is how it starts gaining its clonal expansion and gets into malignant transformation. If it is intact, if the P53 mechanism is intact, it will try to repair a cell, and if the cell is repaired, it will be sent back to the system. If the cell is not repaired, it will activate pro-apoptotic molecules. So the moment it backs, that is a pro-apoptotic molecule, the P53 sends a signals to the cell saying that you are defective, kindly produce BAX molecules. The moment the BAX are synthesized into the cell, it automatically signals that it has to undergo apoptosis. And that starts breaking the DNA into pieces and that will undergo a non-inflammatory cell death and the cell will be removed from the system by the internal homeostasis. The other important mechanism of malignancy is like
here in the apoptosis, BCL2. So
BAX BCL2.
Yeah, mitochondria. Whether any research is going on.
There are lots of
that the
BCL2
mitochondrial mutations, whether how mitochondrial mutation contributed to the oncogenesis.
This BCL2, when you ask specifically, these mitochondrial mutations most of the times will be inherited from the maternal cell, that's mother. So there is lots of inhibitory molecules or small molecule tyrosine kinase inhibitors which are being on the process or in the phase of development for this BCL2 specifically. So I'll just come to the that subsequent slides. So what happens is a normal cell which is undergoing a replicative process will have something called as telomere, which will keep shortening over a period of time, and the moment the length of the telomere shortens, it begins to have attained senescence, and the cell will be warded off from the cell system. When this checkpoint, or when the cell to sense that it has attained senescence, if it is lost, the moment the shorter telomeres lose that sensing of senescence, then the moment the checkpoint is lost, they will undergo, if there is an intact checkpoint, then probably they will undergo a proliferative arrest, and from there it goes to apoptosis. The moment they do not sense that the telomere is short, or the moment they do not sense that there is no senescence, there is a checkpoint, the single checkpoint is being bypassed, and thus undergo a multiple replication process which causes genetic instability and malignant transformation. So all this is about clonal gain. I mean, like how do they acquire genetic mutations? What is the molecular pathogenesis that is being involved in this cell for a normal cell to become an abnormal cell and for that to become a malignant. The moment a cell is designated to be malignant or it has been deviated from the normal process of cell cycle, it will start replicating its own clones. I mean, the way normal cells were being replicated is totally different from the abnormal cells because they have no death signals which are coming. So if they are uninhibitedly, they have an uninhibitory signals where they start replicating in an uninhibited way, and they start expanding their clone. And the moment they start expanding their clone, they start creating a microenvironment from where the invasion of the cells happens. So say this is the basic cell lining in the basement. The moment they start replicating, they start sending signals to the adjacent cells and extracellular matrix. And again, this microenvironment is something which is being researched a lot of late when immune modulation of cancer has started playing some picture in control of malignancy. So what happens is extracellular matrix, the adjacent extracellular matrix of the individual cells would start breaking down, and that would allow an easy access of the malignant cells to other places. From there, they start their journey from there, and they get access to the blood vessels. From there, they get home to another place of their blood circulation, and they start expanding their clones where they get deposited again. So a cell which has started at one place gets an easy access to the vascular supply, vascular medium or blood vessels. From there, it gets home to another place, and the place where it gets home, it starts replicating again, and that is how it invades and metastasizes to other places. This is one beautiful picture I could pick up showing an entire molecular pathway which is being involved in cell cycle. The growth signals which are getting the growth factors getting attached to the cell membrane, growth factors, from there the signal transducing proteins, and from there to the nuclear regulatory proteins and cell cycle regulatory proteins. You can go through this. Just to give a brief picture about this is all about the cell cycle which is happening at the molecular level, and anything which is any damage or any hazardous effects which are happening in the cells from an environmental perspective, say a carcinogen, be it a chemical carcinogen or a radiological or virus mediated. This carcinogen would undergo a metabolic activation to cause electrophilic intermediates, and these intermediates would undergo detoxification process if the cell if the cell is able to handle it appropriately, it will be taken off from the system. If not, these electrophilic intermediates will start binding to that of the DNA and causing DNA adducts. These DNA adducts in terms convert into a pre-neoplastic clones and then to the neoplastic clones and final formation of malignancy. This is all about the basic picture that how a cell gets transformed to a malignant cell, and on a cross note level of how micromolecular organisms are completely imparting on a cell cycle and its growth. The major challenge, like when we see all this, the major challenge in controlling the tumor cells was that we should be able to target only the tumor cells. At the same time, we should be able to conserve our normal cells too. So that it's striking a balance between the tumor cells versus the normal cells is where the majority of the times we get the gain out of it. To begin with, what happened is the moment we started introducing treatment strategies into the system, be it chemotherapy or radiation therapy, the way they act is, be it chemotherapy or radiotherapy, they would act on rapidly multiplying cells. Say, if entire tumor cells will be made up of bunch of cells, some of the cells will be in the quiescent phase, some of the cells will be in the G1, some of S, some of G2, and some of them are in M phase. So it is completely a mixture of cells in a different phases of cell cycle. Whenever we try to give chemotherapy or radiotherapy, they can act only on the synthetic phase of few of them, most of them. They would act only on the S phase of the cell cycle. In that way, rapidly dividing cells will be targeted than that of the than that of the other phases of the cells of the other phases of cell cycle. Say, today I have around 1 kg of tumor. Imagine that there is somebody who is going through some sort of treatment modality. I have 1 kg of tumor, but out of that 1 kg, around 500 grams of that tumor cell would be in the S phase, and another 250 would be in the G1 phase, another 250 in the M phase, another 250 in G2 phase. If I give some chemotherapy today, it would go and act only on the S phase of the cell cycle, or radiation would go and act only on the S phase of cell cycle. It wouldn't be acting on rest of the phases. So, say if I give chemotherapy only today and stop it, what happens is only the S phase of the tumor cells are targeted, and rest of all those which are there in the other phases of the cell cycle would definitely come back to the S phase over a period of time and would take it as a signal where there is a damage which is happening, and they start replicating in a double way than was happening before. So what we learned out of this is like sequencing chemotherapy and radiation therapy. That is when we learned that this tumor duplication time, we tried sequencing chemotherapy and radiotherapy in such a way that the moment the next cells are recruited into the S phase, we would strike again with the chemotherapy or radiation therapy. That is how we try to sequence chemotherapy and radiotherapy. Say, if we are trying to give chemotherapy, it would be either weekly or once in 15 days or once in 21 days, depending on the tumor, depending on its duplication time. Similar way, radiation therapy, where it is not the entire dose of radiation is not given on one single day. It is given in a fractionated way. It is small amounts of radiation which is deposited every day, so that we try to target only the S phase of the cells every time. So, moment today, if I give some amount of radiation, tomorrow again few cells will be included in the S phase. So, this is how I try to target cells, the entire bunch of cells of malignancy every day. This is how we tried evolving from understanding the cell cycle of the tumors and sequencing our radiation and chemotherapy into the cancer treatment. As I told you, the striking a balance between when I try to give chemotherapy and radiotherapy, they do not have a notion that whether it is a normal cell or it is an abnormal cell. It would go and act on all the cells in the body, be it normal or abnormal. Abnormal cells, the malignant cells will be impacted more because more number of cells in the malignancy would be in the S phase compared to that of a normal cell. So chemotherapy does not have an idea whether to act on the malignant cells or only on the normal cells. So in that way, if it acts on the normal cells, we would definitely come out with side effect profile, and definitely any chemotherapy which we try to impart would have a side effect profile. So to strike a balance, to understand it much better, what we tried is we get into in depth about cancer genome, just to know that is there anything that we can only target the cancer cells, avoiding the normal cells. So that is when we started knowing cancer genome. The basis of this cancer genome is, as all of you know, that the genomic mapping are redesigning the tumor taxonomy by moving it from one histological grounds to that of the genetic base level. To give an example for this, what I say, how do we label a diagnosis is, say it is carcinoma lung, what is the histological variant? Histological variant is based on the cell, whether it is a squamous cell or adenosquamous cell, based on that. So we were at that level where we were just trying to label it in a histological way, and we have moved from there to that of a genetic level, where now we try to impart some sort of a genetic alterations at the specific level, so that we can personalize the medicine depending on the individual patient. The success of cancer drugs designed to target the molecular alterations underlying tumorigenesis has proven that somatic genetic alterations are legitimate targets for therapy. As I told you that we understood that there are some sort of genetic somatic mutations which are happening at the micromolecular level, which is imparting a malignancy. So that is when we try to analyze in depth to know what somatic mutations are happening, and is there any molecular target that we can design so that we can block the mutations which are happening or which are producing some self-sufficient signals for the tumor to grow. In that way, we can personalize the medicine to an individual patient. This tumor genome technique is helping clinicians to individualize treatment by matching the patients to the best of their treatment options. Tumor-specific DNA alterations are not every tumor would have a similar genetic makeup or similar genetic mutations. In that way, having an individual genomic cancer genome would definitely help us in tailoring the treatment to the individual cancer patient. Ongoing analysis of multiple cancer genomes will definitely add on to additional targets to that of treatment TT for us to target in a personalized way. Any idea what it is? So identification of cancer genes. As I told you, we learned that there are some somatic mutations which are happening for a cell to have a malignant transformation. Microarrays. Transformation. And we tried to know what sort of genomic transformations which are happening. The major landmark for this is a human genome project which completed in 2004, which gave a complete genomic picture of a human DNA. As of 2015, I see there is some 30 gaps which are still there in that, and they are not able to complete that. But nevertheless, whatever the human genome project, which gave us a complete picture of nucleotide sequence of entire DNA, that led to a drastic research in terms of malignancy, especially in terms of cancer care. All this, I'll just leave it to you. Probably you can go through the slides later. I can just say that the Human Genome Project is one major change which was brought in to us to know what is a nucleotide sequence, and based on that we started developing our molecules which can really target the specific mutations which are happening in malignancy. So this is the upper panel. What you see is that of histological changes, that is, which are happening at the cellular level, the morphological changes, what we see microscopically, and this is the genetic changes which are adjacent to that. If you start comparing both of them, this is that of a colonic cancer. If you see, this is the intestinal cells. The moment these are the normal epithelial cells. The moment there is a ABC beta catenin gene mutation, the normal cells will become dysplastic cells. You can see this this is normal cell, and you can see there is a loss of the normal nuclear pleomorphism, and that it starts picking up pleomorphism and it becomes dysplastic. So the initial change of this ABC beta catenin, the downstream pathway, I'm just trying to picture the downstream pathway from the signaling pathway to that of the molecular changes which are happening at the patient level or the individual level. So normal epithelium picks up the dysplastic. Dysplastic is something which is an abnormal cell, but it's not malignant. So this dysplastic cell would acquire further mutations in terms of KRAS, 18q and P53 would land up in metastasis. And with subsequent sets of mutations which are happening with the downstream pathway, say APC beta catenin mutation would definitely land up only in dysplasia, not amounting to malignancy. The similar cells which are already having an APC beta catenin mutation would acquire further mutation in KRAS gene, from where the dysplastic cells would start moving into an early adenoma. Early adenoma is a phase where they are pre-malignant. So there are two mutations already now, APC beta catenin and KRAS. So a cell which has acquired two mutations, APC beta catenin and KRAS, would acquire another mutation in 18q, where the moment it acquires, adenoma cell will start moving into later stages of adenoma, like early adenoma, intermediate and later adenoma. It's just a phase of cell going through malignant transformation. The moment it has acquired 18q, it is already there in the late adenoma phase, and the cell which has acquired all three mutations and gained another mutation of P53 would end up in malignant transformation. So it is not a single mutation which is causing a malignant transformation. It is a kitty of mutations which are happening from all through the signaling pathway and ending up in malignant transformation. The similar morphological changes I just depicted just to say, if we can block one phase, probably a cell cell picking up a APC beta-catenin pathway, if we try to block somewhere there, then probably for a cell to acquire further mutations and to get converted to malignancy would be stopped. So that is what I wanted to convey with this picture. With the Human Genome Project and with the complete mapping of the human genome, what we did next was like we all know that the single sequencing and then the mutational R's of the exomes, and now we are in the phase where complete exome sequencing is what we are looking into. Exome is something where the transcriptional DNA, complete transcriptional DNA, to know that the entire transcription. I will let you know why do we need to know the entire transcriptional DNA in depth. So cancer genomes have an average of 30 to 100 somatic alterations per tumor. As I told you, the somatic mutations which are there, APC beta-catenin, then acquiring P53, then acquiring 18q. All this, they have around 30 to 100 somatic alterations per tumor coding region, in the coding region, which is the exome region, which has a higher number than that we have previously been thought of. So knowing this exome or coding region of the DNA is very important going further to tackle the malignancy in a better way. Even within the single cancer type, as I told you, the colonic cancer, a single colonic cancer can have multiple somatic mutations and a significant amount of tumor heterogeneity. You mean to say a certain clone of within the colonic cancer, a certain clone of cells would harbor a different somatic mutation compared to that of a different subset of similar clones. This concept of individualized tumors having a unique genetic value is highly relevant to this era of personalized medicine. As I told you, the spectrum of this nucleotide context of mutations differing between tumor to tumor type. For example, 50% of the mutations in colonic cancer are CG to that of a TA transitions, and that of 10% is only CG to GC. So knowing these exomes or this nucleotides at the transcription level is important because 50% of the patients will be having that of CG to TA transformation as compared to that of CG to GC. The reverse is true in terms of breast cancer. That you mean to say that all the malignancies couldn't harbor the same nucleotide sequence is what I'm trying to convey. At the same time, knowing this nucleotide sequence would definitely impact in future research and at the same time to having specific targets. This knowledge of this mutational spectrum is very important as we can develop personalized medicine to individually to a patient. Solid tumors arising in children, especially the medulloblastoma, it's a kidney tumor, which harbor around 5 to 10 times less genetic alterations compared to that of an adult type. It is the same medulloblastoma, same brain tumor, which is happening in pediatric group and that of an adult group, but harbors a varied amount of genetic mutations. That means that they do not behave the same way, even if you have labeled it as the same malignancy. So these pediatric tumors harbor fewer applications and homologous deletions within the gene pools and compared to the pediatric. So initial sample sequencing was a little difficult. That is when we tried developing next generation NGS, next generation sequencing, where there are lots of platforms which are available to have NGS, which makes our life very easy to know the entire gene mutations which are happening in an individual patient with an individual malignancy. These are just the platforms, how they function, what is the template they use, how they extract, how they sequence, what is the iteration and the DNA that they can do. And I have just validated it, you can go through later. As I was telling with that colonic cancer, the pathway directed where the growth factors, cell receptors, signal transmitting proteins, nuclear regulatory proteins. So initially what we tried doing is with the genomic mapping, we tried to have a cancer mapping where patients' genome is completely mapped and we try to know there are multiple amount of mutations which are happening. The moment we try to collecting data is easy, but we interpret is really difficult. So when we have so many mutations which are happening, we didn't know what mutation is acting at what level. There are certain mutations which are helping the tumor to grow, there are certain mutations which are helping the tumor to metastasize, there are certain mutations which are helping the tumor to get resistance to treatment what we are already trying to do. So we did not know at what level these mutations are trying to act on the tumor cell. So that is when we started reworking towards pathway-directed approach, where we try to complete the entire genome of a malignant cell and try to label it. This is how it would look in terms like what mutational spectrum, what are mutation rate, methylations, RNA expression, pathway analysis, structural variations, copy number variations, and mutation profile. So if I have a patient and if I do a complete mapping of that patient in this similar way, then probably I would definitely, easily know that where exactly I'm going wrong. Probably where is that I can act, what is that I have to do. We were like, as I told you that we might have identified lots number of somatic mutations. Say, I have started a patient on a treatment and I have modeled multiple number of mutations and I've got a target molecule to block certain mutation. But if I do not know what is that the mutation is helping to the cell, then it's of no use. Say if a mutation is trying to help it to attain a resistance and I'm trying to give the molecule or a medication, it's of no use. There's no point. So to know that the entire pathway directed mapping is what we are looking into. Probably if a patient comes to me and if I have this pathway directed mapping, then probably I know that where exactly I have to act. So where exactly I can block the molecule, design a molecule so that I can block the pathway and arrest cell cycle growth or biogenic transformation. There's a new taxonomy of tumors where this is the 100. These are like 100 patients of colonic cancer. As I told you that not all the 100 patients would harbor the same somatic mutations. So a few of them would harbor only KRAS, BRAF. These all the mutations as you go through. Probably as you start learning more about it, you will understand. So few of them would harbor only KRAS, few of them would harbor only BRAF. So few of them would harbor only P53. So the entire colonic cancer is a blanket term. At the same time, not all the 100 patients of colonic cancer will harbor the same somatic mutations. So developing an individual patient-based cancer genome profile definitely help us to understand where exactly the patient fit in, because all this what I'm trying to talk like KRAS, BRAF, whatever I'm trying to talk about, we will have a molecule designed so that we can block this mutations in terms of helping the patient. Say I do not know.
Here in colon cancer, it's a multi-step process, right? So whether it is a KRAS or BRAF, any P53 mutation only, you could
Definitely. The moment
you make out it is a
yes,
colon cancer.
definitely. But the moment is, the thing is, P53 as a molecule would require for a normal cell too. So to develop anything against P53 is little tricky. To know what sort of a mutation P53 is acquiring is important. So as you told, it's definitely a multi-step process, and the theory what says is like if the cell is trying to acquire mutations in a step level, probably stopping it at the first step would definitely help it, leading to a P53 mutation is what we are trying to look into. So in that way, this picture would definitely make sense to develop such individualized or personalized medicine. Probably what we used to think is like all these 100 patients giving a chemotherapy would respond in the same way. No, all these patients wouldn't respond the same way because all of them are having different somatic mutations, and it might be the same chemotherapy, but the response might be different because their somatic mutations are behaving in a different way.
So in continuation to that, sir, so we have like first step, second step, something like that. Like, do we have a whether they have categorized these mutation could be of first step, or whether it could
No, that is what. As we say, as I told you that the growth factor receptors, signal transcription proteins, tyrosine kinases down the lane, and the nuclear regulatory factors. They have labeled the factors, they have identified the factors, but we are not able to pinpoint and say this could be the reason. That is when we started having this pathway-directed approach, and Sanger is the one place where they are trying to develop this mapping and bring it to the picture so that probably we can identify that specific molecule and act there. So we do not know. It is completely multifactorial, and where downstream pathway we are trying to hit, we really do not know.
Well, depends on the driver mutation, which is the driver mutation which is responsible for hemolysis. So if you target the driver mutation, then there are chances that passenger mutation can take its seat over the driver mutation. So that's how the disease progresses even after one line, second line of treatment. Then targeting those pathways is important.
So here
As rightly pointed out, the moment we try to look into the genomes, probably we have lots of passenger mutations along with the driver mutations to know that this is the driver mutation, and if we are successful to develop a molecule where we can block the driver mutation is where the success behind the treatment lies completely.
So here in the colorectal cancer especially, what happened, the first generation, like the person is infected, even he has the thing. So whether you counsel that his progeny, the chances are very, very high in breast and colorectal cancer.
We do this genetic.
It all depends on the NGS. Nowadays we are getting all patients with NGS now. So whether the patient is having a somatic mutation or a germinal mutation. If the patient is having a germinal mutation, then there is a need for genetic counseling, family counseling, so that early detection and screening methods and all. If the patient has a somatic mutation, then treating that patient and counseling him and further treatment process continues.
So such germinal mutations do exist in breast and prostate and endometrium and colon. And of late, BRCA is one which is probably in breast when it comes to genetic counseling. Of late, many people are showing interest when it comes to breast cancer.
But as the HER,
Also, he had talked about it.
that is commonly used.
No, HER2 is a protein actually.
Protein they will.
Epidermal growth factor receptor. In some patients, HER2 will be over-amplified. So it will be, as Vinay pointed out, like it will be a continuous stimulation for cell growth. So in those patients, HER2 will be over-amplified. So targeting those HER2 receptors, blocking those HER2 receptors, will stop the malignant growth, further malignant growth. So there are so many drugs which are anti-HER2 agents, is blocking those HER2 receptors.
Again, the HER2 may not act similar way in all the malignancies.
Okay.
So that's
Initially it was like HER2 will be only there in the breast. But now there are so many studies where HER2 is amplified not only in the breast, in biliary tract cancers, in lung cancers, in colon cancers, in pancreatic cancers. That is the reason why there is a complete genomic data of the patient is important, because we won't be knowing which gene is overexpressed or which gene is activated. So that we can target that gene. If a patient with biliary tract cancers is having HER2 overexpression, giving him anti-HER2 agent will have a more efficacy than giving him random chemotherapy.
Just a continued part of it. I'm like what I was talking all about the treatment aspect. When it comes to knowing the genetic profile or the DNA alterations or the mutations, somatic level mutations, is there anything that we can pick up the malignancy at the earliest, or is there anything that we can bypass the biopsy? When we try to say there is a tumor or there is a growth, a swelling or an ulcer, to confirm histological diagnosis is very important. Mean to say we'll have to take a part of the tumor, part of the growth or an ulcer, and that has to be put under the microscope just to document it as malignant. For that single tumor cell to become evident as a tumor or as an ulcer, as I told you, it will undergo a multiple replicative process, and the moment it is in a full-fledged tumor stage, that is when we try to diagnose malignancies. Is there anything that we can do at the earliest? I mean, even before a cell has picked up its malignant potential, I mean, a single cell, is there anything that we can do? Is what we can do something called as liquid biopsy. Liquid biopsy is where we try to extract the circulating tumor cells and the tumor cell DNA. If we know, so suppose if I know that it is a lung cancer and these are the genetic somatic mutations which can happen in a lung cancer, if I already have this idea, then probably doing this liquid biopsy where I can just draw blood sample from a patient and analyze the cells in the blood. If the cells in the blood carrying the similar profile of somatic mutations which are already known for that of a lung cancer, then probably I can easily say that the patient might develop lung cancer in near future. That is where we are heading towards liquid biopsy, where knowing genetic or somatic mutational pattern in a specific tumor would definitely hint us in early diagnosis. Not only that, suppose if I have this part is about all about early diagnosis, where I can pick up malignancy even at a single cellular layer than that of a full-fledged tumor. At the same time, if I have imposed a patient on certain sort of a treatment, and if I want to know, because most of the times what happens, as I told you, that we are trying to act on the S phase of the cell cycle, and we are really not sure that we have attained the zero cells when I'm trying to treat malignancy. say if any single cell which is left behind after my entire treatment process would definitely come back as malignancy, that we call it as recurrence. So that recurrence, suppose if I have already treated and that I want to know if there is any single cell left behind, even for that treatment monitoring process, if the liquid biopsies pick up at the earliest, then probably that is when I can really say, okay fine, there is something which is left behind, something we have to act upon, rather than just waiting for it to recur and come back again. So liquid biopsy is something which has been picking up of late in terms of diagnosis and monitoring the treatment, assessing the recurrence patterns, and imparting screening. I mean, there are lots of screening modalities which are available in terms of early diagnosis of malignancy. Whatever we say early diagnosis, it is only at the radiological level or the clinical level. It is not at the micromolecular level. So liquid biopsy is something which can act at the micromolecular level, where I can really say even at a single cell level that the patient might harbor a tumor cell and would end up in certain malignancy if I know that the genetic makeup of that individual malignancy.
So is it in practice?
Liquid biopsies, we do it for monitoring purpose, but not diagnosis and screening of. As of now, I didn't know that ctDNA, if you can do in cervix and pick up this, the screening what we have for CS cervix is only through pap smear. And what I was thinking is if you can do ctDNA even before that is what we were trying to do.
Yeah, I have 4 students, no project students, genetics, okay? So they wanted to take up it. I have not assigned the problem.
So this liquid biopsy is the future, probably, which would definitely.
Could we try as a pilot step?
A module we can think of.
This is it will be
How-
really breakthrough.
How would my future look like? Probably even all this of late, I mean, these days, if a patient comes to me, then probably I will ask for a biopsy just to know what is the histological diagnosis, and I will ask for some imaging modality just to know whether it is at the same place or it has metastasized to another place. Based on that, I will label it as some certain stage of a tumor and I will try to treat it. So the future of this malignancy is totally different now. The moment the patient comes to me, probably in the near future, we would assess the tumor, take an informed consent. Then I would go for the first step would be a tumor genetic analysis or genetic mapping. With the mapping, I would point out, figure out certain somatic mutations. If the molecules are targeted, then probably I'll just put him on that personalized medicine where a specific molecule which is acting at the somatic level, somatic mutation level. And if I have anything to assess with the somatic or germ level mutation, based on that I'll try to segregate my patients and try to treat them with the specific molecules. At the end, yeah.
At the end, because 4 students, they wanted to take up the topic. So at least even I could interact with the students. We went to 2 people. Some pilot, we'll try, we'll see what will happen. So then, accordingly, if it is a promising, then we can write up a project. That's really, they need always some pilot study. They don't need any project, no, simply they don't CMR, they don't. If you have some paper audit. So
there is any guided old nanoparticles now they're using.
Old nanoparticles for
nanotechnology.
For the third generation sequencing is what they talk about.
Okay.
Other than that, I don't have an idea.
The treatment aspect we are using nanotechnology because of increased binding and increased crossover with the segment.
Okay.
Because whatever the chemotherapeutic agents has got a high molecular weight and cannot protein bound and they cannot bind to the proteins and they cannot cross the segment. So that intratumoral concentration of the drugs will be less. So these drugs for better penetration, for better intratumoral concentration, these were tagged to the nanosomal particles, just like albumin. Okay, for example, one drug called paclitaxel is there. So conventional paclitaxel is different. Nanosomal or albumin-bound paclitaxel is different. The difference is that nanosomal albumin-bound paclitaxel has got a better efficacy on the tumor cells and lesser side effects. So it is being used for many drugs like nowadays liposomal irinotecan and nanosomal. Many chemotherapeutic drugs have been tagged with this nanotechnology.
some of these rich nanoparticles,
Gold
gold or silver, they use many. Which one?
Gold is specifically, I don't know. As like nanotechnology is definitely gold.
But which one, which particles they make for more than any
Gold,
Silver.
We are not rich.
Okay, okay.
Gold specifically.
Okay. So questions from students. You have shown the wind pathway also, and this one, Manohar. So the pathway reducing catering and particle. Proteins.
One last question. Say a normal person is there, this is a normal habit, is going on, okay, he reaches 50, 60, and suddenly he develops the lymphoma. What is the reason behind?
At this point of time, I can only say it is multifactorial. We really cannot pinpoint and say this could be the only reason that he has developed malignancy. This genetic makeup over generations we might have already acquired. This present generation might have already acquired mutations which we do not know. So we are trying to know what exactly is that. We are at the micromolecular level. Those mutations can only answer. Probably the entire genetic makeup of this generation is already altered. That is how we started witnessing large number of malignancies in this generation compared to the previous ones. That is how, as we say that this acquiring genetic or somatic mutations happen over a period of time. It is not an overnight journey. Probably generation to generation we have acquired mutations in such a way that our entire genetic makeup is already at a threat now. Probably a small sort of an injury, even at the stress levels, would end up in malignancy. So entire genetic makeup itself is being damaged is what I feel.
The liquid biopsy, how much accurate?
How much?
Accurately is the result?
That's what. To know accuracy probably in terms of treatment, like monitoring the treatment strategies, there are certain evidences which says that we can really correlate. But when it comes to screening and diagnosis, we are really not sure about it yet.
That's what the sir wanted to check. If it is good, then not.
Basically, liquid biopsy depends on the tumor load. It is just like there is a solid tumor, we do biopsy, that is a solid biopsy. Liquid biopsy taking out the blood and looking for the tumor cells or circulating tumor cells or circulating free DNA. If tumor load is too high, tumor cells are very high in the circulation, then the accuracy is low. So definitely it's not comparable to the regular biopsy.
Sir, but could be the first line of screening. If they find the chance more low,
The cell-free DNA.
No, screening liquid biopsy is not a modality as of now.
That's what I'm trying to tell you. If I know that this patient of lung cancer has these many amount of somatic mutations and if these mutations are causing lung cancer, these are the driver mutations, because of this the patient has developed lung cancer, then probably I can keep these somatic mutations as my markers and extract the cell-free DNA from all the normal population and try to analyze or try to have the genetic makeup and match with that of malignancy and try to know, okay fine, these mutations are there, then probably this person might end up in lung cancer near future is what we can predict. So we are not there at that point.
so like a pap smear test in the cervix. So if we develop a small thing like first line of screening, where but this.
The thing is, for pap smear, we are trying to extract the cells from the site, exactly from the site. The cervical cancer happens at that place, and we are trying to extract the cells from there, and definitely we would witness some sort of variations. For the tumor cells to dislocate from the site, get into the bloodstream, and extract the DNA from there, we would definitely need the genetic makeup, which is already happening.
Because it will be a very localized disease.
Yes.
By the time it comes to circulation,
It will be metastasis condition. Yes.
So we still do not know. So probably that is where the research should happen. Is what
Yeah. Some pilot studies, if they are promising, we can continue, otherwise we could drop it with those ideas.
Even for monitoring purpose, there are all the malignancies. Even if we treat today, and I cannot declare them that you are cured of disease.
Yeah.
There is a threat that the disease might come back. The recurrence might happen anytime.
Yes.
And is there any simple monitoring technique that we have? Can we do ctDNA just as a main monitoring of this treatment aspect is also a good thing. That can change the picture.
So regarding the expression profiles, so when we do NGS, at one point of time we are taking the sample and we are doing the sequence and then further analysis. But I don't know whether there are expression profiles, okay, in first stage or whatever the initial stage.
Definitely, that might be different again. As the entire tumor set would go to multiple stages and the same somatic, that's what I'm trying to tell you. We do not know. Is it the same somatic mutation which is causing the similar picture in all the patients? Do not know. The similar somatic mutation might cause the cells to duplicate in one patient, might cause cell arrest or resistance to the treatment in other patients. So we do not know. To know that this is the reason for this patient, then probably we can personalize the medicine saying that okay fine, you can target this somatic mutation.
Excuse me. So chemotherapy and radiation therapy mainly focused on treating the cancer cells which are present in the S cycle. So is there any treatment or technique which can be cure the cancer cells which are present in other phases, that G1, G0?
There are chemotherapy molecules which can act, what we call it as cell cycle non-specific chemotherapy molecules. But the cell kill, what happens, it will be definitely more in this phase of cell cycle compared to that of the other phase of cell cycle. So we do not have any other things that, even if I give radiation or chemotherapy, the target population will be S phase. Definitely rest all the phases of cell cycle could be impacted, but not to an amount to that of an S phase.
So what could be the reason, like, there's a lot of prevalence these days with respect to cancer, like globally as well as in India. And the medications which are being given are way too expensive and people cannot afford it. Like, most of them cannot afford it. So, what would be your opinion on cutting the cost of medication?
Logistics, probably the simple platinum molecules what we used to use 10 years before were costing thousands, 1,500. Now they have dropped down to 250. Probably going forward everything would logistically definitely because give a yellow on the sequence mat like the cost what we impact would be definitely transferred to the patient.
Yes.
So from their gross level to the patient level definitely be impacted. Probably as we go forward, probably more and more research would definitely compact and tone down this logistics over time. Only time can answer.
Thank you very much, sir.
Recently I have
We'll finish off the other talk also and then we'll have another session.
recently I came across an article from the Cell. So they showed us carcinogenesis as a species. So what is your point on that?
Carcinogenesis as
Species specific.
specific.
No. That is leading to a new species. So why should we consider that?
Speciation.
No, I did not get the question. What do you
For the example,
No, the whole process of carcinogenesis should be considered as an evolution of a new species. So that's what they
new species with malignancy.
Yes.
So
some resistant
the answer, I seriously don't have an answer. Logically, if you talk, probably some decades back when diabetes was the major disease or major non-communicable disease, and that was a threat at that time. Probably we have started living with diabetes over a period of time. I call it as an evolution now. Probably we have learned to live with diabetes. Similar with the evolution, what speciation, what you are talking about, is probably over a period of time we try to live with cancer or malignancy. That's what I can say. Like I don't see any evolution out of it. Probably we try to that becomes a chronic disease, and we adopt it for that, and we try to start living with that. Is what I feel.
It's like malignancy, more or less with new research molecules, it is becoming like a chronic disease. Just like initially when CML came, chronic myeloid leukemia, there was no treatment, and once imatinib came into the market, like it is TK, tyrosine kinase inhibitor. Now people with CML are living for 10 years, 15 years with that tablet. And also in lung, CLN, because of these mutations and all, there are tablets which are coming out. With these tablets, people, well, 4-stage patients are living for 5 years, 6 years. So as time progresses, newer molecules will come up and more or less as it were to become like a chronic disease.
Now I request our chairperson ma'am to listen to words.
Just 5 minutes more to come.
Now we will break for 5 minutes. After that, everyone are requested to ask the guest speaker.
Good afternoon to all and welcome back to the second session of today's special talk. It is my privilege to introduce Dr. Kiran P.K, an expert in the specialized field of medical oncology dealing with the assessment, diagnosis and management of cancer patients. Dr. Kiran P.K is an expert in the field of medical oncology with an MBBS degree from JSS Medical College, RGUHS Mysore, a gold medalist for Best Outgoing Resident in MD in General Medicine, SN Medical College at Dr. B.R. Ambedkar, Agra, Uttar Pradesh, and a DMP in Medical Oncology from National Board of Examination. With over 9 years of work experience in the field of medical oncology, contributing 3 years as a DMP resident in medical oncology and 1 year as a senior registrar and consultant medical oncologist, he is currently appointed as Assistant Professor at JSS Medical College and Hospitals, JSSAHER, Mysore. Dr. Kiran P.K has also been involved in nursing undergraduate teaching and training. Dr. Kiran P.K has attended various CME programs and conferences on recent advances in medical practices and medical oncology, reflecting his broad interest in oncology and infectious diseases. He has published research articles in national and international journals with his significant topic of dissertation being to study the prevalence of vascular complications at the time of diagnosis in a newly diagnosed asymptomatic type 2 diabetes mellitus patients and their relation with various risk factors, comparison of peripheral blood mononuclear cells with CD34 positive stem cell apheresis yield and correlation to the duration of neutrophil and platelet engraftment following autologous stem cell transplants. Their profound understanding, coupled with a tireless commitment to patients and a drive for pioneering research, has solidified their position as a leader and innovator in the battle against cancer. As we prepare to glean insights from Dr. Kiran P.K's wealth of expertise, let us acknowledge their unwavering dedication to advancing medical oncology and let their tireless efforts in research, teaching, and compassionate patient care serve as an inspiration to us all. I once again welcome Dr. Kiran P.K. So, thank you.
So, I'm confused with the good morning or afternoon. It's already past 12. Thanks for that wonderful introduction. It's always very to hear about yourself, so much things, what you have done. Regarding today's topic, I'm very glad to be here. Thanks, Madam, for the opportunity. and I hope you people are ignited now, because we are also at your level unignited or not ignited when we are students. The right time, because of teachers like Malini Madam, we are here now. So thanks to those teachers. And regarding today's topic, good introduction has been given by Dr. Vinay regarding carcinogenesis. I hope all of you have got a good idea about how the molecular pathways and how carcinogenesis is produced, and how the tumors will develop and metastasize and become stage 4 from stage 1 tumors. So my topic is quite a broad topic, quite a vast topic. It's difficult to cover it up in a short session, in a single lecture. So I'll try to just brush up whatever is important in as simple language as possible. I hope most of you know about some part of immunology, and then brushing about the immunotherapy and how is it related to cancer, and some of the newer drugs which are coming up targeting these immuno cells in the cancer. So this is once again, I think you are not bored with this slide. Once again, the same normal cell division where there are close to the apoptosis here, and uncontrolled check of the uncontrolled proliferation of the cells leads to tumor. That is malignancy. Now, whenever we talk about cancer, there are 3 varieties of cancer. One is carcinoma, one is lymphoma, and one is sarcomas. So it all depends on from where it originates. If the cancer originates from the epithelial lining of the cells, then it is called as carcinoma. If it is arising from the hematopoietic origin, like blood cells, RBCs, WBCs, then it is called as lymphomas or lymph nodes. And immune system and comes like spleen, lymph nodes, bone marrow. So all this comes as lymphomas, myelomas, and leukemias. If it develops from the soft tissue, muscles, bones, then it's called as sarcomas. So majorly oncology cancers is divided into 3 different groups: carcinomas, lymphomas, leukemias, and myelomas. Those are hematopoietic cancers, and this is sarcomas. So being a medical oncologist, I treat carcinomas, leukemias, and lymphos and myelomas to some extent, and sarcomas. Majorly, this hematopoietic origin malignancies are treated by hemato-oncologists. Okay, this is for just a general knowledge for your information. Then coming to the immunity. So there is a basic, let's brush up some basic terms about the immunity. There are 2 types. One is adaptive immunity and one is innate immunity. Innate immunity means one which is there within the body. Adaptive immunity, once you acquire either from outside, either from the mother or either from the injections, immunizations, and all. So adaptive immunity can be either natural or artificial. Natural by passive, that is maternal, where the antibodies get transferred through the placenta from the mother to the fetus. That is natural passive immunity. Natural active immunity by infections, repeated infections. Most of you all know, like children's, neonates, infants get repeated infections because they don't have this natural adaptive immunity, except for maternal. So by acquiring these repeated infections, they get immune, they will get antibodies, they will get resistance for those infections, and so adults don't get as much infections as infants. So in basic terms. And artificial immunity is passive immunity, that is antibody transfer. We give this in medical field where we give IVIG, intravenous immunoglobulins. This immunoglobulins will be given in autoimmune diseases, just like I hope you have heard about some Guillain-Barré syndrome or autoimmune diseases and all. There we give IVIG, artificial immunoglobulin transfer, and active, that is immunization. Once we give for the kids like BCG vaccination, DPT vaccination, tetanus vaccination, all this. This is artificial active immunity. Okay, so basically innate and adaptive. Innate, once again, natural and artificial. Natural is mother, artificial is by passive antibody transfer or by active immunization. Now coming to the same, what's the difference between the innate immunity and the adaptive immunity? Innate immunity is the first line of defense. Suppose as soon as enemy enters enters your house, the first line of defense is innate immunity. That is rapid first line of defense. Then it has got specificity to antigen, no, very low specificity because whatever comes, if it's an antigen, it's a foreign body, it fights. There is no specificity, there is no diversity of response, there is no potency, there is no memory. Suppose if the same enemy encounters again during the second or third time, it doesn't know. So once again it attacks like a new enemy. That is innate immunity. Whereas adaptive immunity takes time to build, just like antibodies to the antigens. It has got high specificity, high diversity of response, high potency, and has got memory. So once there is a secondary infection, you don't get any symptoms of the disease because of this adaptive immunity. Because they have memory cells, memory B cells, which can attack the secondary antigens, and then it can build an immune response. Now coming to the parts of immune system, basically there are mucous membranes is also part of immune system, nose and tonsils, thymus. Thymus is one important thing, is located here behind the sternum. Thymus, where the T cells get activated. There are basically two cells in the body, T cells and B cells. T cells get activated in the thymus and B cells get activated in the bone marrow. So here is the spleen, once again the maturation of the cells, and bowel, the lining mucosa, and also in bowel there are small intestines and large intestine. In the small intestine there is one part called ileum. In the ileum there are patches called Pierce patches where the lymphocytic aggregates will be. So that also acts as an immunity system. Then comes the bone marrow, where most of the hematolymphoid cells get originated, replicated, and maturated. Then skin. So bone marrow is very important since all the blood cells, T cells and B lymphocytes originate here. They get maturated here, and B cells remain in the marrow to mature, whereas T cells travels to the thymus and there they get maturated. Now, regarding the T cell development, because we are speaking more about the T cells today, so this is the T cell development where T cell precursors will be from the marrow goes to the thymus here. From the thymus, there are two types of T cells will be generated: CD4, CD8 plus, and CD4 plus. CD8 is called cytotoxic T cells, where it kills T cells. It is the frontline defense, just like a soldier. Then CD4 cells is called activator cells. It activates the cytotoxic T cells. So when it comes to CD4, once again these are divided to 4, that is T helper 1, T helper 2, Th17 and T regulatory cells. What you need to remember is CD4 plus is activator cells, helper cells that won't kill directly. It activates the CD8 plus T cells which attacks the antigens. So how the CD4 activates the CD8? Through these mechanisms, through this interleukins and chemokines and all like interferon gamma and interferon. One important here is this T regulatory cells. Not all CD4 cells activate the CD8 cells, but there is one more cell, T regulatory cell. It reduces the immune system. Why this is important? Because there needs to be a balance between the activation and regulation. Because if the T cells keeps on activating, at one point of time it attacks body's own cells. So to prevent the T cells from attacking body's own cells, these T regulatory cells are there, which inhibits this activation. So this is how the immune system functions. Innate immunity is the first line of defense, where the immunity components are natural killer cells, basophils or granulocytes, lymphocytes, monocytes and all. And here the adaptive immunity where T cells, CD4 T cells, CD8 T cells and B cells which produce antibodies. So these are specific, attached specifically to diverse stimuli. B cell antibody production will be there, T cell stimulation, and also important thing is memory functions. Now the same slide, natural killer cells and cytotoxic T cells. Cytotoxic T cells means CD4 or CD8? CD8. So CD8 cells, cytotoxic. Memory cells means B cells, which keeps the memory. And this is the suppressor cell. This is called what I told, T regulatory cells, Tregs. Okay, Tregs suppresses the immune system. So once again, this how this gets methylated: CD4, CD8, NK cells, T regulatory cells, plasma cells, and also the macrophages. Macrophage 1 and 2. Now, what happens when there is an antigen which comes into the body? There are cells called as antigen-presenting cells, which are also called as macrophages or dendritic cells. This detects the antigen here. These are called as antigen-presenting cells. This engulfs the antigen and processes the antigen and makes it into small pieces of cells called peptides. Peptides means proteins. Peptides. Here, it engulfs here. Phagocytosis, by the process called as phagocytosis. It processes this antigen where antigen is degraded and present as peptides. So what happens after that? After the peptides, these dendritic cells, means antigen presenting cells, will have this major histocompatibility complex receptors. So this is very important because this MHC is very unique to individuals. So once this peptide is processed, through this MHC, this is a peptide here, this peptide comes over this MHC and gets attached. It comes outside and it's attached. This is recognized by the T cell. This T cell is recognized through this TCR, means T cell receptor, and then it's get activated. So there are three various mechanisms of activation of T cells. One is either this MHC TCR complex. Second one is CD8 or CD22. This is one more receptor which acts between these dendritic cells and the T cells. And other route is by secreting interleukin 12, this T cells gets activated. So dendritic cells is the main processor of the antigens. It makes the antigens into peptides and presents through its MHC receptors to the T cells, and T cells gets activated. So this T cells, activated T cells, acts only on this peptide. This is specific. This is called as adaptive immunity. It doesn't act on other antigens. It acts only this processed antigen which is there. So this is just pictorial diagram where this CD8 cytokine killing cells in squamous cell carcinoma where the 1 minute and this phagocytized, and here the tumor cells is killed.
So is it real-time
Yes.
Yes.
That is the time taken by the T cells to kill the tumor cells. Now, with all these basics of immunity like T cells, what is its relation to the cancer? Basically, in cancer treatment, there are 4 types. One is radiotherapy, immunotherapy, chemotherapy, and targeted therapy. First discovered was chemotherapy. Chemotherapy is the one which treats the cancer for all the era since World War II. It was mustard gas, which nitrogen mustard was the first chemotherapeutic agent which was found to be active against lymphomas. Then so many other chemotherapeutic drugs came to market. As Dr. Vinay mentioned, chemotherapeutic drugs cannot differentiate between the normal cells and the cancer cells. That is where the major disadvantage of chemotherapy. That is the reason why most of the chemotherapy drugs has got major side effects. And chemotherapy drugs acts on highly active cells. That's what the picture was shown in the last presentation, where most of the cells which are in the synthetic phase, S phase or M phase, will get destroyed. In our body also, like gastrointestinal mucosa, hairs, and this oral mucosa, all these are actively divided. Daily our mucosa will be shedding off, daily our hairs will be shedding off, so these are actively divided. So whenever we give chemotherapy, along with that tumor cells, this also acts on these normal cells. That is the reason most of the chemotherapy drugs have got these side effects of hair loss, oral ulcers, and death. Because of these cells are also get destroyed along with the tumor cells. Suppose we can imagine just like a field, soil, which is our body, and plants. These are the normal cells. Some weeds will come up in between. These are the cancer cells. So, surgery is like completely removing the weed from the route. Along with that, some other normal cells will also be taken off. And spraying over some plain or grown, that is called chemotherapy, where normal plants will also get affected and weeds will also get affected. Okay? Then along with that, radiation means spraying only in some parts where more weeds are there. That is radiation. So normal cells will also be, normal plants will also get exposed. But targeted therapy is the one where you go to plug a single weed. That's what Madam was telling about anti-herpes agent, EGFR agent, where a particular genetic mutation is possible for the production of cancer, where we are going to target a drug which can target that gene or that protein which is overexpressed, that is called as targeted therapy. Now what is immunotherapy? Immunotherapy means increasing body's immunity so that the specific T cells get multiplied. So that is called as immunotherapy. Now where do you put this immunotherapy in this whole concept of soil, weed and plants? There are some fertilizers where they don't allow a weed to grow, or whatever weed is grown, they don't allow it to nourish. That fertilizer is added to the soil, that is called as immunotherapy. Okay? So in a simple concept, this is what immunotherapy does. It acts through the soil, it acts by using body's own defense mechanism. That is called as immunotherapy. So why immunotherapy is important? Why it is chosen? Because of its specificity. It can precisely attack the tumor cells and potentially clot the body, thus minimizing damage to the surrounding tissue, unlike chemotherapy. It is long-lived. So once it is given, the duration, once the tumors are responding to the immunotherapy, the response is longly durable. It is like if we keep on giving the immunotherapy, the tumors will keep on responding. Then third one is adaptable. So it will be effective in different types of cancer, regardless of the specific tumor, because we are boosting body's own immunity. So immunity has got nothing like this cancer I should act, this cancer I shouldn't act like that. So it has got adaptability and of course memory. Once again, adaptability has got memory. So by all these features, immunity means immunotherapy has got severe target molecules inside the body. So targeting immune system is the newer concept for treatment of cancer. Now this is the cancer and immune system, where these are the hallmarks of cancer till 2000, where Dr. Vinay told in detail about wasting the cell death, inducing angiogenesis. Angiogenesis means production of new blood vessels. Angiogenesis, new angiogenesis, where the tumors get nourishment from newer blood vessels and enabling replicative immortality, activating tumor invasion, metastasis. So all these things are the hallmarks of cancer till 2000. But after that, when this concept of immunotherapy came, then avoiding immune destruction. So ability to escape of the immune response is now recognized as a hallmark of cancer. So with all this immunity in the body, with all these T cells, still why cancer is growing? What's the reason for that? Because we are activating T cells, we have got inside body's immune response, innate immunity, natural killer cells, T cells. Still, why cancers are growing? Because cancer cells are cheating this immune system. Okay? They are cheating this. So how they are cheating this? I'll come to the next slide. And so, this, if we can block this cheating mechanism, then we can able to treat cancer properly. So that is what the ability of escaping the immune response is now recognized as hallmark of cancer. So this is the immune response to cancer. The genetic and cellular alterations and characteristics cancer cells and recognized by immune system as a foreign, resulting in T cells being able to kill the cancer cells. This is the first stage. As soon as the cancer cell, just like foreign body, it activates the T cells in the body. The entire process by which immune system recognizes targets and kills the cancer cells is described by, that is called as cancer immunity cycle. Now, tumors can inhibit the anti-tumor response. This is important because tumors can inhibit the anti-tumor response by disrupting the balance governing the steps of the cancer immunity cycle through a specific pathway or a combination of pathways known as immune checkpoints. So this is how the cancer cells cheat the normal cells, cheat the normal T cells. How? Now here, this is normal cycle. Once the cancer enters in the body, this is the cancer cell. Antigens are presented. Dendritic cell, as I showed in the last slide. This dendritic cell is presented with T cell, which activates, multiplies. The same specific T cell multiplies. It infiltrates in the tumor and then recognizes the cancer cells and attacks the cancer cells. Now, by this mechanism, cancer should not grow. All cancer cells should be died, should undergo apoptosis or whatever, phagocytosis and all. But what happens here? This is the immune escape mechanism by the cancer cells. The first immune escape mechanism is poor immunogenicity of the tumor. What if the tumor cells does not express antigens itself? Then the dendritic cells cannot be knowing what is the antigen. Okay? Then, so reduced dendritic cell maturation. Dendritic cell will remain stable, it remains standstill because it won't get the antigen to process and to convey it to the T cells. Suboptimal T cell activation. So, now here, suboptimal T cell activation. Now the second is immune escape is improved T cell trafficking and filtration. So impaired T cell trafficking and filtration. Once, even though if this T cells gets activated, this trafficking is impaired. That means T cells cannot go to the proper area or T cell cannot multiply themselves because of checkpoint inhibitors from the tumor cells. These checkpoint inhibitors are developed by the tumor cells itself. This prevents the T cells from multiplying and thereby attacking the tumors. Now, one more immune escape mechanism is that the interference with immune recognition and killing of the tumor cells is impaired. So, tumor cells, T cells are activated, T cells are multiplying, but they cannot kill the cancer cells. So, cancer cells has got this ability to escape from this normal immune mechanism. So, immunotherapy targets these areas where the immunogenicity of the cancer cell will be increased and T cells trafficking will be increased and the recognition of the cancer cells by the tumor T cells will be increased. So, loss of MHC function, that was MHC is a receptor on antigen presenting cells, that is dendritic cells. So, if that is lost, then no antigen presentation or identification. If overexpression of checkpoint inhibitors, then that also no cytotoxic cells. Immunosuppressive microenvironment, just like increased T regulatory cells. T regulatory cells inhibit the immunity. So, if there is increased T regulatory cells, no response. Now, this is the tumor microenvironment.
How that T regulatory cells 17 will be inhibited? Which are the clues or stimulus?
No, that's when T regulatory cells will be increased. Increased. This will inhibit the immune system. T cells
And
inhibit the CD4
clues
cells.
for the changes in the
Yes, no, changes in the microenvironment and different chemokines and cytokines. For example, interleukin 10, TNF beta, interferon gamma, all this will be secreted. That thereby inhibits one more like MDS2, MDS myeloid-derived stem cell factors. All this will be secreted by the T-reg cells and also by the tumor cells. So this is the tumor microenvironment. There are so many targets, so many drugs can be developed with this tumor microenvironment where CD8 cells are there, MDSs, T regulatory cells here, as you know here, T regulatory cells, this express CTLA4. So there is a drug, I will come to it later, there is a drug which can block that CTLA4 because T regulatory cells by expressing the CTLA4 receptor inhibits these dendritic cells. So there is a drug which can block the CTLA4 and thereby keeps activating these dendritic cells. That is the reason why tumor microenvironment is very much important. Now, in a simpler way, this activated T cells will be inhibited by tumor cells by PD-L1 receptors. I will come to PD-L1 later. And MDSC cells, macrophages, activated T-reg cells through interleukin 10 and TGF beta, and dendritic cells by PD-L1. So all these cells inhibit this T cells. So everything will be from the tumor. Now, how compiling all these mechanisms, how this tumor cells escape from the immune system is one is elimination. This is the first step where the T cells will dominate here. In the elimination,
T cells will proliferate and tries to eliminate the cancer cells. This is called as elimination by different molecules here. T cell activated T cells, CD4 cells, CD8 cells, everything will be here. As you can see here, the tumor cells are also here, and this is the dormant tumor cell. Now comes the equilibrium. Here what happens, the anti-sensitivity of the tumor cells reduces. So whenever the tumor cell gets to know that somebody is attacking, it becomes dormant. It reduces its antigenicity so that it do not expose itself that I am your enemy. Okay, so here what happens, the T cells will also become little suppressed because it doesn't recognize a tumor cell because of low antigenicity. This step is called as equilibrium. Next comes escape. So when the T cells are dormant, when the T cells become inactive, the tumor cells, once dormant, it becomes further, now it comes to know the characteristics of T cells, how the T cell is getting activated, what and all is happening in the T cells. The tumor cells gets activated and produce certain molecules, produce certain receptors which can block this activated T cells, and thereby it grows out. So here we can see number of tumor cells got engaged. So the
Are there any parallel evolution wherein, because this you can see lot of thing by this cancerous cell, tumor cells. So parallelly, whether our body is to develop many mechanism, just it hides, no, as you mentioned. So are there
that is where the challenge is.
evolution?
That is where the challenge is.
Naturally, are there any such things? No.
Because T cells are the whole body mechanism, short acting, antigens. So when T cells are suppressed, there is nothing. So the three major steps of this immune escape is equilibrium, elimination, equilibrium, and escape. These three major steps. This is what happens to human body when the cancer cell is there inside the body. Now comes immunotherapy. So where is in all this concept of this immune system, T cells inhibiting and cancer cells proliferating, where is your immunotherapy goals? So immunotherapy goals are, one is it is anti-tumor response, immunosuppression, it reduces immunosuppression, means whatever the immunosuppression because of the tumor cells, because of the Treg cells, because of the interleukin 10, MDS, everything, and also increase the immunogenicity of the tumors. Okay, whatever the hide and seek from the tumors, it tries to uncover it. So from where they started, a little bit about the history of immunotherapy. This was Colis toxin, that was the first immunotherapy in 1989 was there. So he was the one where Colis toxin mixed bacterial vaccine. He injected this toxin into a patient with some skin cancer here. So after injecting, the tumor burden has reduced. So from there, the first WHO approved drugs in immunotherapy as one is rituximab. Rituximab is a chimeric antibody, chimeric receptor antibody. One CD marker, I don't know whether they know about CDs, cluster differentiation markers, CD markers. CD20 is one which is overexpressed in B cell lymphomas. B cell lymphoma. So targeting this CD20 is rituximab. Rituximab is anti-CD20. It is a chimeric. Chimeric means mouse and human, both combination. So rituximab is the one. Trastuzumab is anti-HER2. As I told you, HER2 is one of the protein which is overexpressed in some of the malignancies. So targeting this HER2 will lead to suppression of cancer and cancer cell death. So rituximab and trastuzumab, these are the WHO approved first immunotherapy. More than immunotherapy, it is called as trastuzumab is called as targeted therapy. It targets only HER2. Okay. Now this is the history of immunotherapy from 1863 to 2014 till here. We have first FDA approval anti-PD-1 melanoma, that is some other drugs called nivolumab. My later slides have the drug names and all, and how does it work. So whenever we come to immunotherapies, these are different types of immunotherapy. Whether it is non-specific immune stimulation, that is cytokine injections, interleukin 2 injections. Interleukins are injected to increase the immunity, that means stimulative interleukins. There are suppressing interleukins also, but stimulative interleukin injection can also be given First it was given in RCCs, renal cell carcinomas, interleukin 2 injections. Now no longer it is used now, because when we give interleukin injections, the side effects body's own immune system, just like increased autoimmunity, increased destruction of own cells happens. That is called a cytokine release syndrome. So that happens, very difficult to manage. So that was not that is not used now. Then infectious disease vaccine, just like BCG vaccine is given for bladder cancer. For localized bladder cancer, BCG vaccine, local infiltration of BCG vaccine is given. Then dendritic cell approach, like sickle cell T, which is used in prostate cancers, and oncolytic virus therapy. Some viruses which are active against, just like viruses are active against human normal cells. Some viruses are engineered so that they will be injected, and which are active, they go and kill the cancer cells. That is called as oncolytic virus therapy. And PD1 receptor inhibitor. PD1 receptors are expressed on the T cells. Basically, this activated T cells express PD1 receptors. So those receptors and CAR T cell therapy. This is a new thing, CAR T cell therapy. So we go in detail one by one. 4 major. One is vaccination, adaptive cell transfer, antibodies, and oncolytic viruses. So cancer vaccines. Cancer vaccines can also be given, but the problem is it is not therapeutic. It's only prophylactic. There are cancer vaccines which are available for CS cervix, which is only prophylactic but not therapeutic, because once the virus is there inside the body, the vaccines cannot control it. That is the reason the exact means the proper age for giving CS cervix vaccines is before a person becomes sexually active. So once a person becomes sexually active, then he acquires HPV infection. So before acquiring HPV infection, if you give, then only it will work. That's why these are more or less prophylactic, not therapeutic.
So any other cervix, any other vaccines are there?
Still, so this is dendritic cell vaccines. This is non-specific vaccine, dendritic cell vaccine. And this is cellular therapy, adaptive injection of cells. What happens is, whenever there is a tumor, obviously there will be activated tumor cells. So we collect this T cells, activated T cells, collect it, grow in the lab, replicate it more, so that in the body tumor cells are not allowing them to replicate inside. So we take these T cells, replicate it more in the lab, and then re-inject it. But while collecting, we won't be knowing these T cells are activated T cells or normal T cells, because we cannot cherry pick each T cells. When AFS is done, these T cells will be taken out, they are replicated, and then they will be re-injected. So we infuse the tumor-specific T cells in the patient. This is a picture where tumors, after giving this T cell therapy, after 22 months of therapy, the tumor cells here in the liver. This is the liver. Multiple this shady areas. These are the tumor cells. This is disappearing. So, as I told you, that's the disadvantage. We won't be knowing exactly what T cells. So, what if there is a way to make all the T cells reactive against cancer? So, whatever T cells we are receiving, all T cells, can it be tumor specific? So, that is where when this CAR T cell therapy comes. CAR T cell therapy means chimeric antigen receptor T cells. What they do here is they will take the T cell from the body, tumor specific T cells, single T cell, tumor specific T cell. They inject this CR. CR is just like a tumor antigen, exactly for the tumor, which is specific for the tumor. Chimeric antigen, this receptor will be given to the T cell, and that T cell becomes a CAR T. CAR T means chimeric antigen T cell. That receptor, this will be grown into millions, this will be engineered and replicated. Then it is infused. So all these T cells are very tumor specific. Because of the receptors, they go and directly act over the tumor cells. That is called as CAR T cell.
This is recombinant DNA
Yes. So this is called as CAR T cell therapy. Right now in India, some major centers like in Mumbai, Tata and all, they are using CAR T cell therapy. Basically, this CAR T cell therapy have been used in lymphomas first. Okay. So these are the drug names right now in market, not in India, but in the world, somewhere in major it is very difficult to pronounce it. Okay, so there are different, so this is the target antigen, this is the CAR antigen. Just like I told CD20, just like CD19, CAR T cell, CD19 and this also CD19, BCMA, other things. So most of them are lymphomas, Hodgkin's lymphoma, ALL, mantle cell lymphoma, follicular lymphoma, all this, and multiple myeloma. CAR T cell therapy is basically useful because the important thing is this won't affect the normal cells. At the same time, because of increased T cell, because of increased immunity, side effects are more. Just like
But
side
solid
effects.
tumors,
Not yet.
not yet. This is for the lymphomas.
Because for solid tumors, it's very difficult to get that antigen, exact one antigen and one CD marker. Yes. Now coming to oncolytic viruses, so let's give cancer a cold, just like we get cold. Let's give cancer also a cold. He is Dr. John Bell, oncolytic virus pioneer, and this is what they do: genetically modified virus, which acts only on the tumor, which is injected. Is first it is increased means GM-CSF. This is colony-stimulating factor, which increases the T cells and B cells. Colony-stimulating factor. Then it also gets a systemic response. Why? Because tumor, as I told you earlier, tumor reduces its immunogenicity by not exposing its antigens. But whenever this oncolytic therapy will be given, this tumor cells will explode. So whenever it explodes, more number of antigens will be exposed to the body, and it also has got systemic tumor-specific response and death of the distant cancer cells. Now in October 2015, the USFDA approved the first oncolytic virus therapy to melanoma. This is injected, not IV, directly to the tumor. So the virus is genetically modified version of herpes simplex virus. Is called as TVEC. That is calimogene aspect and side effects. Once again, fatigue, fever, chills, nausea, blood injection side effects, deficiency reactions. Now coming to the antibodies. Antibodies, as we know, like Y-shaped molecules produced by the B cells, basically bind to highly specific antigens, and this can be used for a therapeutic purpose. So this is one important feature where there are inhibitory receptors regulate the immune cell activation. There are this is the T cell where multiple receptors are expressed over the surface. Among that, many are stimulatory, many are inhibitory. So always there is a balance between stimulatory receptors and inhibitory receptors. Tumor cells take this advantage of blocking these stimulatory receptors so that the T cells remain inhibited. So these are the inhibitory receptors where PD-1, important thing is this. Two receptors are important, one is PD-1 and one is CTLA-4. So PD-1 is expressed on the T cell, whereas PD-L1 means ligand, programmed death receptor 1 expressed on tumor cells, sorry, on the T cells, programmed death receptor blocking ligand expressed on the tumor cell. So the tumor cells comes and bind this, block this PD-1. When this PD-1 is blocked, the T cell stimulation will be blocked. So there is no further activation of the T cells. So what if we block this PD-1? We block this interaction between PD-1 and PD-L1. Then the T cells will remain continuously activated and thereby act on the tumor cells. Like the same, as I told you earlier, CTLA-4 is one receptor which is expressed on the dendritic cells. Here, the T cells, it cannot activate it because some inhibitory receptors on the T cell blocks the CTLA-4 receptors.
So by modulating this one, the death receptors, whether we can, are you not modulating the apoptotic body?
But in this, because of that only, side effects of immunotherapy comes. Because of that, everything comes with a price.
Yes, exactly.
So these are called the checkpoints. These are called checkpoints, activation of T cells and checkpoints. So basically, these are activating receptors, all these are activating receptors, and the CTLA-4, PD-1, all these are inhibitory receptors. Important one is PD-1 programmed red cell protein receptor and CTLA-4 receptors. PD-1 receptors bind to PD-L1 ligand on APC or the tumor cell. CTLA-4 receptor bind to B7 ligand on APC tumor cell. So thereby inhibiting the T cell. Once they bind, they inhibit the T cell from acting against the cancer tumor cells, thereby leading to growth of the tumor cells. Now, we need to be clear about where the receptors are expressed because it is quite confusing. Whether it is expressed on the tumor cell, whether it is expressed on the T cells, whether it is expressed on the APCs, antigen presenting cells. So always remember PD1 is expressed on B cells, tumor infiltrating lymphocyte, that is on the T cells basically. PD-L1 is expressed on the tumor cells. PD-L1 means ligand, expressed on the tumor cells, whereas PD1 is receptor which is expressed on the T cells. PD-L2 is also there, one more receptor which is expressed on dendritic cells, macrophages and lymphoid cells. CTLA4, as I told you, is expressed on T regulatory cells, but CTLA ligand which blocks the CTLA is expressed on dendritic cells. Now this is the same mechanism. Once again, when PD1 binds to PD-L1 receptor in the tumor cell, it basically tells T cells not to attack. So ultimately, tumor cell not attacked by the T cells. So some cancer cells have large amount of PD-L1. That is where this is the target. So whenever we give immunotherapy for this PD-1 or PD-L1, how we know that this immunotherapy works? This can be PD-1 or PD-L1 can be used as a biomarker. If tumor is having both PD-L1 levels, then the chances of response to this tumor cells to immunotherapy is quite high. So this is a diagram of once again PD-1 and PD-L1 interaction. This is SCL1 is so anti-PD-L1 targets the PD-L1 receptors. Here anti-PD-1, that is drugs, agents binding to PD-L1 restoring anti-tumor activity. There are drugs in the market where we can block the PD-1 and PD-L1 and CTLA4. These are the three important drugs: PD-1, anti-PD-1, anti-PD-L1 and anti-CTLA4 antibodies. This is once again the CTLA4. These are the drugs: nivolumab, pembrolizumab are anti-PD-1, anti-PD-L1 are atezolizumab and durvalumab, anti-CTLA4 is ipilimumab. So whenever we give these drugs, it blocks this interaction between PD-1 or PD-L1, CTLA4 or BA, and then it keeps continuous stimulation of T cells. Now, these are the, as I told you, response to immunotherapy is whether we don't know because all these immunotherapy drugs are quite costly. This costs around 1.5 lakhs, this costs around 2.5 lakhs per injection, this is 4 lakhs per injection, this is 2.5 lakhs, and this is 2 lakhs.
How many doses they have?
At least for the response, because as you know, this is adaptive immunity, just not like chemotherapy. It takes time to build the immunity. So we have to give at least 4 to 6 cycles before we know the response. 6 cycles minimum 6 cycles. But in a stage 4 cancer, if we give, there is nothing like time limit. We need to keep on giving them.
It comes to 25-30 lakhs.
It will be difficult in India because most of the people don't have the health insurance in India. But in US, Canada and all, everything comes with the government health schemes.
The BP in the
No, the thing is, all the ABRK and BPL schemes will have this chemotherapy. The basic chemo drugs will be. Even what I told you, the first initial drug, rituximab and trastuzumab and all. Rituximab is covered under BPL. Trastuzumab is not covered. Even that costs around some 10 to 15,000 per injection. That is also not covered in the BPL.
15,000 and 1,000, okay.
Yeah. Earlier, I'll tell you, someone asking about logistic question.
Yes.
What's the cost and
Yes.
this thing. See, the thing is, this will be prepared with lot of deep dive and deep research and all. So companies will have a patency for those drugs. I'll tell you an example. As CD4, CDK4 was discussed, a patient in breast cancer will have an increased CDK4. So, if this CD can be targeted by drugs, then that is a treatment for estrogen receptor positive breast cancers. That is, drugs called palbociclib and ribociclib are there, which can target the CDK4 molecules. So, this palbociclib, once it came, it was 80 to 90,000 per month treatment. 1 month was costing around 80 to 90,000. Now, recently, last year, that patency got over. Now it's available for 3.5%. Okay, so all middle class families and even lower class families can afford for that. So time should tell. Maybe this immunotherapy is in the next 5 to 10 years, it will also become cheaper and it will be available for general populations. So this is the area of research, as I told you. Knowing the response, what are the biomarkers of immunotherapy treatment? Means whether this treatment will work or not. For example, in breast cancer, we will check for estrogen receptors, HER2. If HER2 is positive, then only there is a point in giving anti-HER2 treatment. So what's the biomarker for immunotherapy? Like that, PD-L1 levels, PD-L1 expression, tumor mutation burden. Tumors having increased antigenicity can expose themselves to the tumor cells, therefore increased immune response can be mounted on those tumor cells. If the tumors are having reduced antigenicity, slow growing tumor, then it cannot be, immunotherapy cannot work on those cells. These are called as cold tumors. Tumors with low antigenicity are called cold tumors. Tumors with increased antigenicity, increased antigens, increased tumor mutations are called as hot tumors. So there is a way where we can turn these cold tumors to hot tumors by combining this immunotherapy with either oncolytic therapy, oncovirus, where virus is injected and it makes tumor to die and release of antigens, or increasing this tumor mutation by giving chemotherapy, a small dose of chemotherapy. So there are studies going on where combining this immunotherapy with other agents which turns this cold tumor to hot tumor, so that the immunotherapy will work. That is the research area. And also biomarkers, where routinely will do this tumor mutation burden, PD-L1 expression, PD-1 expressions, and genetic pathways, antigen tumor load. Of course, once again, tumor microenvironment and circulating factors, ctDNA, circulating tumor DNA. So all these are biomarkers. Once again, when we give immunotherapy, always there will be side effects because of increased proliferation of T cells. So patients having already increased immunogenicity like autoimmune diseases are not candidates for this immunotherapy because it will flare up. So whenever we have autoimmune diseases, then steroids should be given to suppress the immunity. But here by giving immunotherapy, once again we are increasing the immunity. So patients will have a lot of side effects like dyspnea, cough, but all these side effects, the incidences are very low when compared to chemotherapy drugs. Only less than 1% of patients will have pancreatitis, uveitis, demyelination, autoimmune neuropathy, Guillain-Barré syndrome, hypopituitarism, and myasthenia syndrome. Rest are all, most commonly we have fatigue, we have loose stools, we have pneumonitis, we have mild cough and all, but easily manageable. Some less than 5% of the patients will have serious side effects which can be managed. So the takeaway points, immune system, there are two types, one is innate immunity and adaptive immunity. There are two types of immune response, once again natural killer cells and T cells. Basically T cells are the target for treatment. And there are inhibitory and activating checkpoints present on the T cells, as equal to different receptors, activating receptors and inhibiting receptors. PD-1 is important, which is expressed on the T cells surface. It binds to PD-L1 receptor on the tumor. The T cell action is blocked and cancer cells are not attacked. So checkpoint inhibitors like anti-PD-1, that is nivolumab or pembrolizumab, or anti-PD-L1 like atezolizumab or nivolumab can be given where it blocks this interaction between PD-1 and PD-L1 and thereby acts as a treatment for cancer. Early recognition of immune-related adverse events are vital in managing patients on immuno-oncology. Thank you. Doubts?
Yeah.
There are viruses that cause cancer.
Yes, sir. Many viruses.
So is it available in the environment?
Yeah, like cervix is caused by human papillomavirus. Epstein-Barr virus can cause esophageal carcinomas, where we use some viral load levels, and papillomavirus can also cause head and neck cancers. And in esophageal carcinomas, we use EBV viral load to see the tumor response as a monitor sometimes. So there are viruses. And hepatitis C virus can cause esophageal carcinomas, liver cancers, hepatitis C virus, spreads the virus to some extent.
So how many environment where it is?
It all depends on how it spreads. How it spreads.
Sir, if by chance thymus gland is damaged, sometimes operated, what's the fate of the person?
No, if it depends on the age, at what age it gets operated. Because all this T cells activation, everything happens in the younger age group. Once they become adults, then thymus will get regressed and not much of a work for the thymus because already the memory cells, the T cells, everything are already there in the body. There is no need for maturation of T cells in the body. So if thymus is taken out initially during the childhood, then patient will become immunosuppressed for lifetime. Chances of opportunistic infections are very high. That's why thymus is one of the important organs where in case of myasthenia gravis and other autoimmune disorders, thymectomy is also used as a modality of treatment to reduce the immunity of the patient. Because myasthenia gravis is once again autoimmune disorder.
Sir, HIV patient, whether this, how, if by chance he is HIV positive, so and he
For
wanted to take
immunotherapy in HIV patients, I'm not sure because HIV basically I think CD4 cells once again the T helper cells will be attacked by the retrovirus. But HIV basically retrovirus, the mechanism of action of attacking the cells is quite different with
no, this immunotherapy treatment,
No, that,
they come
yes.
with the HIV positive, if you find that this is HIV positive, and they want you to go for immunotherapy means then maybe you have a lot of challenges. Chances are there. Yeah. This talk is not for chairperson. Okay.
I don't know whether they have understood something or.
No, they understood, sir. They are very, very smart.
No, that I know.
Yeah.
Can the blood vessels that supply nutrition to the cancer cells be targeted by
Yes, very good. Actually, there are drugs which can target these blood vessels. There are receptors. These blood vessels are basically the development of the blood vessels needs some receptors, like VEGF receptors, vascular endothelial growth factor receptors, VEGF receptors. There are drugs anti-VEGF drugs which can target this neoangiogenesis. Okay, and that VEGF ligands which activates this VEGF receptors, blocking this VEGF ligand, like one drug called bevacizumab. It's an antibody basically. So that can be given, bevacizumab and other ramucirumab and other drugs are there which can cut off this blood supply and thereby tumor regresses. But it is an indirect way of treating the cancer. It is nowadays it is like combining chemotherapy with this drug to stimulate further effect. It cannot be used as a single modality of treatment. So there are regimens where, because one cancer is hepatocellular cancer, liver cancer, which is highly angiogenic. Okay, so chemotherapy drugs are quite less effective for this type of tumors. So there is a regimen where we combine immunotherapy with this anti-VEGF, both, and give it to patients. There, the blood vessel will be cut off, increase immunity, and thereby the tumor cells are well suppressed.
And a lot of research is going on to block the VEGF factors.
Many
Major
drugs,
research, they just want to block it.
many drugs are there, and many drugs are already there in the market. Now they are trying to combine these different combinations so that to give an optimal response. And one more thing is all this immunotherapy, what I told, these are nowadays initially when it came, so whatever drugs comes, they try in stage 4 patients. Now gradually then they try with stage 3 and stage 2 and stage 1. Still for early stage cancers, chemotherapy is the treatment of choice. Along with some targeted therapies, immunotherapy is still reserved for stage 4 disease and some stage 3 diseases, just like in lung cancer and bladder cancer. We use it as a maintenance treatment in some cases. But still studies are going on to bring it to the front line.
So whether you practice this immunotherapy in this people are under immunotherapy.
Almost or 5 to 10% maybe. People can afford for immunotherapy because of this insurance and high insurance schemes and some insurance like ESI and all they provide free. CGHS schemes, central government health schemes and ECHS, ex-servicemen schemes. All this immunotherapy is included in this scheme. So they'll get it for free.
And the recovery rate is
It's quite good. As I told you, the duration of response will be more. If at all the tumor is responding, the duration will be more. Once again, it all depends on the immunogenicity of the tumor microenvironment, the antigenicity of the tumor burden low, antigen low.
Sir, anyone going related to this immunotherapy related to that immunology with gut microbes? Because nowadays a lot of results.
Infusing gut microbes.
Yeah.
This is used in I think injecting gut microbes is used in I think IBDs, some irritable bowel syndromes and inflammatory bowel disorders and all is used.
To activate the immunity, so always they say there's a crosstalk between the brain and the gut microbes.
Brain and gut. That is what they say. There are studies, but nothing is proven. So giving probiotics and prebiotics, nothing is proven.
But they are in the initial stage.
What is the status of this HeLa cells now? It is used throughout the world.
No idea.
Huh? You maintain it as a custom.
Still in research, not in the practice.
Okay.
So still in the bench, not as computer said.
Yeah. So, use of immunomodulators alone can help it. Like you said, it is used in combination with chemotherapy for stage 3 and stage 4 treatments and stuff like that. So, how effective will it be like if it is used alone? Like only
used alone.
So you said you are using it in in combination with
Anti-VEGF is used in combination, not immunotherapy. Immunotherapy will be used alone only, but sometimes to make the tumors from cold to hot, there the combination will be used. But most of the times immunotherapy will be given alone. Suppose, for example, I tell you, in lung cancer, we will check for this PD-L1 levels. PD-1 levels. It will be given as a percentage. Some method is there like Venn method. It will be given as a percentage. If the patient is having 0%, there is no expression of the tumor cells, no antigenicity of the tumor cells. If the percentage is 1 to 10%, then immunotherapy can respond. Immunotherapy can act on those tumor cells, but with a lesser extent. In those cases, we use to increase the antigenicity. Immunotherapy is combined with the chemotherapy. If the PD-L1% is more than 50%, then it is highly immunogenic. In those cases, there is no need for chemotherapy. So, single agent immunotherapy is the treatment of choice in CLM stage 4 with PD-L1 expression of more than 50%. So, that is what the biomarkers help us in deciding whether which patients will respond, which patient doesn't.
Sir, can we give the plasma therapy to the cancer?
Hmm?
Can we give the plasma therapy to cancer, like whichever of the cancer arising from the oncogenic virus that are there?
It is plasma therapy is non-specific. It is not specific to the tumor cells.
Some of the cancer arise from like a herpes simplex virus, right? So can we use the plasma therapy?
That's what engineered adaptive T cell therapy. That is same like plasma therapy. Because plasma therapy, non-specific plasma therapy will be given in many autoimmune disorders. Just like Guillain-Barré syndrome and some osteoarthritis and other patients. But it is quite non-specific, cannot be used in this if it is T cell specific. That is what the basic mechanism in adaptive T cell transfer and the CAR T cell therapy.
Because as we told you earlier, other drugs are very costly. So instead of that, can we use this one?
No, that is non-specific response.
So what about CRISPR-Cas? Recently they won Nobel Prize also. So
Gene silencing.
CRISPR-Cas.
Yeah.
So how frequently now they are using it in therapies?
Very recently.
It's upcoming thing.
Nothing is being implicated.
Gene silencing.
Gene silencing.
Gene silencing.
Nothing is happening.
CRISPR-Cas.
They are still in progress.
even Parkinson's and all these things, so they are like.
New things.
Vinay sir, I have one question for you. Be it like tobacco is like the people are spraying a lot of DDT and other kind of pesticides into it, and it's a common thing, like it's involved in smoking, tobacco chewing and other things. So if they don't spray, like say it an organic tobacco, how much can it be a carcinogenic? Like the effects reduce.
Tobacco is tobacco.
Will the effect reduce?
I just wanted to say because
Just like smoke cigarettes, smokeless cigarettes, and some menthol cigarettes and for ease.
Yes.
Many patients used to come. So if I take menthol cigarettes, then will I don't get cancer or the exposure to carcinogenesis. Actually, menthol cigarettes will have more exposure because it causes lesser irritation. People used to smoke more.
so DDT has nothing to do with tobacco. So any pesticides or insecticides, whatever they use, they have nothing to do with tobacco. Tobacco per se has almost like 7,000 chemical compounds which have been extracted, and out of 7,000, 70 are known to be carcinogenic. So tobacco itself is a carcinogen. So there's nothing to do with pesticides or insecticides for people. So tobacco is a carcinogen.
So continuation, sir. So do you have any data of active smokers and passive smokers?
Lots of data is there. There is something called as second-hand smoking, and now of late we call third-hand smoking. So somebody who has been there in and around somebody who has smoked, and they come back home, and somebody who is not smoking, and they can get Parkinson's. Something like third-hand smoking is something which we are already looking at. Lots of data on passive smoking which has proven to be managed.
That is the reason why whenever a patient comes, suppose female patient, village side, come with say buccal tuberculosis, you won't be knowing. See, in cancer there is nothing like etiology. In my own family, my parents and my brother ask, whenever I tell about some patient, they ask what is the cause. So most of the cancers are idiopathic. There are risk factors, not the etiological factors. All are risk factors. So smoking is risk factor, tobacco is a risk factor. So whenever we search for risk factors for the patient, why she has developed this buccal mucosal cancer, then she'll give a history that her husband used to smoke. And then our next question is whether he'll smoke inside home or outside. Because if most of the villages also, people are smoking inside home. So his wife will be by the side of the patient and she keeps on inhaling, and that might be one of his factors. That is the common passive smoking, what we see.
Because much more chemicals will be released through the passive smokers than active smokers. They will be
Even smoking in smoke zone.
That is
smoke.
it will be covered in
Completely covered, and if you smoke one cigarette, you go into 100 cigarettes.
Sir, in case of microenvironment, sir, so there itself there is so much of pressure for the neighboring cells. So before angiogenesis itself, there will be pressure effects will be there and all those things. So before we would identify. So is there any target? Can we target that mechanism?
No, in microenvironment, everything will, tumor microenvironment will develop only once the tumor comes, it proliferates and then takes over the environment into its screen. Then only it comes. Or else the whole body will be in equilibrium. As I told, there are like many genes which are pro-apoptotic, anti-apoptotic, stimulating, inhibiting, everything will be in equilibrium. But with this tumor invasion, that equilibrium will be damaged.
So I request our chairperson to illustrate the test. Take.
Please check.
It's a pleasure for us, sir. visit to Bharat Cancer Hospital. I requested sir, and he is very happy to show all the things. It will be in your syllabus. So, if you know, theory is different and seeing the things is different. So theory is like 4 blind people describing elephant, okay? But once you see how they are doing, it will be really, you will be very much enlightened. And we got an opportunity. Generally, people won't allow outsiders, and on request he told. So yeah, we have a good. He has a good affinity towards the genetics department, and he obliged. And it's a great test opportunity for you to visit, ask question. And one more assignment, each one of you have to observe and prepare for one thing. So for example, PET scanning with the machine, so how do they do or whatever. So each one of you take one assignment and just you can brief. After coming back for 5 minutes, you should explain about that. Okay. So and you will be taken by Dr. Chaitra. Chaitra will be there. Always know we need a ringmaster. Otherwise, okay, you are all academic kids. Generally, we treat them like our kids, and she will be take care today. Okay, with the time. So what time is okay for you? 2:30. 2:30. From there you can move. Okay. So you finish that and then you move. Hardly it will be 1 hour, sir. Hardly it will be 1 hour. 2:30 to 3:30. From there you can. Okay. So Dr. Chaitra will guide you. Okay. Thank you everyone, and thank you sir for the wonderful session. Thank you.
Thank you everyone. I would like to thank our chairperson for giving me this opportunity. Thank you.
ಎಜುಕೇಶನಲ್ ವೀಡಿಯೋಸ್ ಥರ ಮಾಡ್ತಾರೆ.
ಎಜುಕೇಶನಲ್ ವಿಡಿಯೋಸ್ ಥರ
Series should be very compact. ನಮಗೆ reaching medical faculty, general population
ಸರ್ ಅದಾದ್ರೆ ನಾವು ಯೂಟ್ಯೂಬಲ್ಲೇ ಮಾಡ್ಬೋದು. ತುಂಬ ಜನ ನೋಡ್ತಾರೆ.
ಅದನ್ನ most googled question on answer ಕ್ಯಾನ್ಸರ್ ಈ ಥರದ್ದು
ಜನರಲ್ ಡಿಸ್ಕಷನ್ ಸೆಷನ್
ಒಂದು ಬೇಕಾದ್ರೆ ಈ questions are so for that questions
most googled question on the hospital
ಸೂಪರ್ ಡೌಟ್ಸ್
simple ಅಷ್ಟೇ. That is how
Reasons